kindly
Good morning.
deliver
Good
the
morning.
welcome address.
A very warm good morning to all of you. Am I audible?
हेलो हाँ आ गई
Yeah, am I audible? Is my voice audible, Dr. Sumedha?
Yes, sir. Yes, sir. It is audible, sir.
Yeah. So a very warm good morning to all of you. Right now, I'm in South Korea, but this FDP was planned in such a way that it can be on a very contemporary topic. And very big thanks to MNNIT as well as our organizing team at Galgotias University. We have with us our
we are not able to listen the voice.
Dr. Gaurav and Dr. Sumedha and the whole team who have been instrumental in designing each and every aspect of this FDP along with MNNIT team. So a very big congratulations to all of you and a very big thanks to the audience for coming here for such a contemporary topic. And this FDP is going to provide you a lot of things. We are going to have talks on different aspects of smart biomaterials. And as you all know, smart biomaterials have significantly impacted human healthcare by advancing the development of medical devices designed to function within human tissue, mimicking the behavior of natural tissues. So while the intelligence of biomaterials has evolved from inert to active over the past few decades, these smart biomaterials take a step further by making their surfaces or bulk respond on interactions with the surrounding tissues. So this has been very successful in imparting outcomes similar to natural tissue functions. So this interaction with the surrounding tissue helps in creating stimuli-responsive biomaterials, which can be useful in tissue engineering, regenerative medicine, autonomous drug delivery, orthopedics, and much more. So traditionally, as we all know, material engineering focused on refining the static properties of biomaterials to accommodate them within the body without evoking an immune response. Now, this was a major obstacle to their unrestricted operation. But now, the current approaches in research is for developing stimuli-responsive biomaterials that tune their outcomes based on the responses to bodily factors like temperature, pH, and ion concentration, or there are certain external factors as well, like magnetism, light, and conductivity. So these are having lots and lots of applications in soft and hard tissue engineering, 4D printing, as well as scaffold design. So we are going to discuss all these different aspects. In addition to that, we are also having talks related to AI on the relation of AI or how AI can help in designing the smart biomaterials. So we are going to discuss all these things. We are going to have very nice talks over these 6 days, that is from 14th to 19th. And I can tell you one more thing, that the choice of speakers is also very, very good. We are having very good speakers from different parts of the country, and they are from renowned institutes. I would like to thank the management of our university, Galgotias University, for always keeping us motivated and supporting us in all these events like anything. Our Honorable Chancellor, Sir, Shri Sunil Galgotia ji, our Honorable CEO, Sir, Shri Dhruv Galgotia ji, as well as our Director Operations, Ma'am, Dr. Aradhana Galgotia, as well as our Honorable VC, Sir, Shri K.M. Babu, Sir. They have all been instrumental in supporting us through all this, and they have been the motivating force behind this FDP. I would also like to thank the audience which is there, along with the organizing team of MNNIT as well as Galgotias University, for organizing this FDP on a contemporary topic. We have brought a huge lot of speakers for all of you, and let's see how much we can learn. I would like to request the audience to be inquisitive enough, and let's move ahead with good questions, good queries, so that science can move forward. And I would also like to thank, in the end, all the people who have been instrumental in bringing out this FDP with the help of support from IT team. Thank you. Thanks a lot.
Sir, for your thought-provoking speech. I'm pretty sure that the ideas that you have pitched forward in the welcome address itself will resonate in the minds of our participants, and they will think about how to deal with all those things in the future, because yes, it is the future of biomedical research indeed. The smart biomaterials, the stimuli responsive biomaterials, these are the future of our biomedical research. I thank you once again, sir, for agreeing to speak at the welcome address and taking time. We know that you are very busy in the research schedule at South Korea. So if you managed to take out some time for our program, I am really grateful to you, sir, for that.
Thank you.
Thank you, sir. So next, we will move on to our next speaker to set the stage. I request Dr. Gaurav Kumar, HOD, Biotechnology and Bioengineering Department at Galgotias University under the aegis of School of Bioscience and Technology, who is also the organizing secretary of this FDP, to deliver the preamble. Sir, the stage is all yours.
Thank you. Thank you so much, ma'am. Am I audible, ma'am?
Yes, sir, you are audible, sir.
Thank you. Thank you so much, ma'am. Honorable Vice Chancellor, sir, respected Director of MNNIT Allahabad, distinguished guest, eminent speaker, faculty colleague, and dear participant, a very warm and respectful good morning to all of you. It is our great pleasure and honor to welcome you at this faculty development program on smart health innovation, integrating imaging, sensor, and biomaterials. It is jointly organized by the Department of Biotechnology and Bioengineering, School of Biosciences and Technology, Galgotias University, and the Department of Applied Mechanics, MNNIT Allahabad, Prayagraj. This FDP has been co-designed and conceptualized in the collaboration with Dr. Uvanesh K, who is my colleague and my best friend, who has served as a true catalyst in the bringing together the academic minds, institutional synergy, and an inspiring vision for the interdisciplinary learning. We are proud to share that this program has received a response with over 1,500 registrations from across India. I have sent the invitation to all the participants, respected faculty members across the India, and currently more than 450 faculty member has joined our WhatsApp group, and currently approximately 150 faculty member is joining with us. So these faculty member are representing from various NITs, Central University and private university. So this enthusiastic participate reaffirm the growing national interest in the innovation at the interest of the biomedical technology, sensor integration, and advanced biomaterial. Actually, this FDP is structured to highlight the breakthrough concept in the healthcare innovation, ranging from the AI in medical imaging, wearable sensor, bioprinting, transdermal drug delivery to molecular diagnostics, and robotics in medicine. So the academic week is mapped with 4 core UN SDG goal, that is SDG 3, 4, 9, and 17. So let me also take this moment to highlight the remarkable achievement of Galgotias University. The university is NAAC A+ accredited, ranked among the top 50 institutes in India, and recognized nationally and internationally for its excellence in teaching, research, placement, and industry collaboration. With over 98% of the placement success rate and more than 200 MOUs has been signed globally, Galgotias University has become a hub for the innovation and entrepreneurship. Within the university, the Department of Biotechnology and Bioengineering, School of Biosciences and Technology, stand out for its interdisciplinary teaching and research. The department currently having 19 PhD faculty members and offering 3 UG program, that is BTech Biotechnology, BTech Biomedical Engineering, BTech Chemical and Biochemical Engineering, as well as 2 PG program in the MTech Biotechnology and Biochemical Engineering, as well as MTech Biomedical Engineering, with over 327 students. We actively promote the research integrated learning from the undergraduate to the doctoral level and regularly organize capacity building initiative like this FDP. So we are deeply grateful to our Honorable Vice Chancellor, Professor K. Mallikarjuna Babu, and our Chief Guest, Professor R.S. Verma Sir, Director of MNNIT Allahabad, for their continuous support and vision that drive academic excellence across the institution. With these words, I once again extend a heartfelt welcome to all our dignitary speakers, keynote speakers, participants, to look forward for a productive, collaborative, and intellectual stimulating week ahead. Thank you so much. Thank you, ma'am.
Sir, for your very visionary talk and your spirit of organizing this kind of FDP in collaborative manner with esteemed institutions, and which brings to us all the facilities and all kind of resources that we might need for our further development in our career growth, and as well as for increasing our knowledge. Thank you, sir, for this collaborative effort that has brought together lots of stalwarts in the field in the same stage, so that we can educate and progress the knowledge of our peers, and we can gain through peer learning. Thank you so much, sir. Next, we would like to call upon our catalyst for this program, as we like to say, Dr. Uvanesh K. Vishwanathan, who is the FDP Convener from MNNIT Allahabad, so that he can share the conceptual framework and goals of this program. So, Dr. Uvanesh, I call upon you to grace the stage and tell us about the conception of this program.
Thank you, ma'am. Thanks for inviting me. So, Honorable Director, MNNIT Allahabad, Honorable Vice Chancellor, Galgotias University, distinguished guests, esteemed speaker, dear faculty, colleagues, participants, very good morning to all. It gives me immense pleasure to share a few thoughts about this faculty development program on smart health innovation, integrating imaging, sensors, and biomaterials. When we conceptualize these FDP, our vision was to create a truly interdisciplinary platform that brings together scientists, engineers, clinicians, and technologists to reimagine the future of healthcare through collaborative innovation. Today, as we officially commence this academic journey, I am filled with a deep sense of purpose and pride. The intersection of biology, engineering, and digital intelligence is reshaping how we understand, diagnose, and treat disease. From AI-powered diagnostics and wearable device sensors to bioprinted tissue and smart biomaterials. Those possibilities in smart healthcare are expanding exponentially. The objective of these FDP is not merely to update knowledge, but to spark collaborative, provoke new questions, and enable faculty and researchers to bring such cutting-edge innovations in their own classroom, laboratory, and project. Each section in these FDP are carefully designed to represent emerging themes, real-world application from academic laboratory to industrial R&D across India. So I am particularly delighted by fascinated response from the academic community with over 1,500 registration from faculty and research scholars across India, a clear evidence to relevance and urgency of this topic. I extend my heartfelt gratitude to Galgotias University, especially Dr. Gaurav Kumar, who was my senior also. So HOD and FDP programs organizing secretary and the entire team from Department of Biotechnology Bioengineering for their remarkable coordination, energy, and shared vision that make this collaboration with MNIT not just possible but impactful. To all our esteemed speakers who are generously sharing their expertise from NIT, IIT, IIITs, Central University, and industry, thank you for enriching this platform. Your insight will undoubtedly influence the thinking and research of each participant here. So, as the FTP unfold over the 6 days, I urge all the participants to engage actively, ask questions, establish network, carry forward the learning into practice. I am confident that these program will not only ignite idea but also inspire innovation in smart health technology across diverse disciplines. Thank you all for being part of this academic endeavor. Let us together build pathway to healthier and smarter tomorrow. Thank you.
Thank you, Dr. Uvanesh. Thank you so much for
Thank you, ma'am.
presenting so eloquently the vision and essence of this FDP and sharing the conceptual framework and goals of this faculty development program. We hope that this will be really successful as you all have perceived it to be. Thank you so much. I now welcome Professor R.S. Sharma, Distinguished Professor at School of Biosciences and Technology, Galgotias University, to share his thoughts about this faculty development program. Sharma sir, I now request you to kindly grace the stage and talk about your visions and your ideas regarding this FDP. Thank you so much.
Thank you. Yeah. Thank you very much. Can you hear me?
Yes,
Hello?
sir. Go
Yes,
ahead.
sir. Yes, sir. We can
Can you
hear.
hear me?
Yes, sir. You are audible, sir.
Hello, Sumida.
Yes,
So,
sir. We can hear you.
good morning, everybody. I hope everybody is able to hear me.
There is a little technical glitch on Dr. Sharma's part. It is being resolved.
Ma'am, can we move on to Bhadi, sir? Because after that we can accommodate, sir. If he is
Okay.
able.
And
Huh?
we'll fix the glitch.
Huh.
And okay. To fix this technical glitch, I request Dr. Ajay Bharti, Head Officiating at Department of Applied Mechanics, MNNIT Allahabad, to address the gathering. Sir, the stage is all yours. Thank you for taking time off your busy schedule to accommodate us. We would thank you, sir.
Thank you, Dr. Sumedha. May you hear me?
Yes, sir, we can hear you.
Yes, sir. Go ahead, sir.
Yes, sir.
Okay. So, good morning to all dignitaries, organizing team members, and participants. Today, being a visiting head, Department of Applied Mechanics at MNNIT Allahabad, and as a host of department of this program, it's my pleasure to address this inauguration function on faculty development on smart health innovation. It's being organized in between 14 to 19 July. As we know that FDP is being organized jointly by Department of Applied Mechanics, MNNIT Allahabad, and Galgotias University. So, respected Professor Ramasankar Verma, Chief Guest of this function, Chief Patron of this program, and Director MNNIT Allahabad, Honorable Vice Chancellor, Professor Dr. K. Mallikarjun Babu from Galgotias University, Professor Abhimanyu Kumar Jha, Dean School of Biosciences and Technology, Galgotias University, who is co-convener of this program also, Dr. Sumedha Mukherjee, FDP coordinator, Dr. Gaurav Kumar, he is heading the department as well as he is organizing secretary of this program. Dr. Uvanesh, he is very active person of our department, and probably as Dr. Sumedha has already mentioned that he is the catalyst for organizing this program. So he is the convener of this program also. Dr. Saurabh Bharadwaj from Applied Mechanics Department, MNNIT Allahabad, who is one of the coordinator of this program. Distinguished speakers, guests of this program from reputed institutions of our country like IITs, NITs, IIITs, and other centrally funded institutes, research institutes, and industries. Participants attending this program from different parts of our country and media persons. As a department at MNNIT Allahabad, we are 24 faculty members and we are offering two BTech programs. Number one is engineering and computational mechanics. Number two, materials engineering. Similarly, 4 MTech programs are being offered, degree offered. First, maybe MTech, material science and engineering. Number 2, materials, MTech biomedical engineering, MTech fluid engineering, and MTech engineering mechanics and design. So especially Dr. Uvanesh was appointed to look after the responsibilities of running this biomedical engineering program smoothly, and he is actively participating and doing his job very satisfactorily. I will not take much time to talk about the theme of this program because other people who are expert of this area will be speaking in detail. There are few things which are being emphasized here only. While somebody is talking about the smart health, suddenly question arises in our mind, what is smart health and innovation? So therefore, as we mean, a smart healthcare and innovation are transforming the medical field by integrated advanced technologies to enhance patient care, improve efficiency, and promoting the preventive measures. This includes the use of artificial intelligence, IoT devices, big data analytics, and cloud computing to revolutionize the diagnostics, even treatment and patient management. So there are some key aspects of smart healthcare like enhanced diagnostic and treatment, personalized and preventive care, improved efficiency and accessibility. Similarly, patient empowerment, focus on prevention. And you may see the examples of these smart healthcare technologies like wearable sensors, AI-powered diagnostics, telemedicine platforms, electronic health records, big data analytics, data security and privacy, integration and interoperability, and ethical considerations also. So with this, I will stop myself speaking about the theme. However, I assure all the participants that you will definitely will enjoy this program because one can find this program is well equipped with the different aspects of smart health and innovation. So thanks for showing your interest in this program and listening to me. All the best and have a nice day. Jai Hind.
Thank you so much. We thank you for your valuable insights regarding preventive healthcare and personalized medicines, as well as for highlighting the ethical concerns regarding these aspects. We also thank you for your academic encouragement and telling that we should be learning about all these things as we grow in the field of healthcare research. Thank you so much, sir, for sharing your views with us. I now welcome Professor R.S. Sharma, Distinguished Professor at the Department of Biotechnology and Bioengineering at Galgotias University, to share his thoughts regarding this faculty development program. Sir, kindly take the stage.
Thank you. Thank you. Thank you. Good morning to everybody. Let me take this opportunity to welcome all the faculty members who are participating in this conference. I would like to first thank all the authorities of the Galgotias University, the Chancellor Sir, the CEO Sir, the Vice Chancellor Sir, the Dean of the school, and the Dean, the head of Department of Biotechnology. Now, for this workshop or this faculty development program, why it is so important, as we know that so far the healthcare system is concerned, there are 3 key issues are there. What are those? How quickly a doctor can reach to the patient, how quickly the patient can reach to the doctor, how quickly the doctor can diagnose the disease, and how quickly the doctor can make the appropriate treatment to the patient. Now, in this whole process, smart health innovation is playing a very, very important role, particularly in the era where the healthcare is rapidly transforming through the convergence of the engineering, the biology, and the digital technology. This programs comes at a very, very important time at this stage. So the integration of the smart imaging techniques, advanced biosensors, and biomaterial is redefining the diagnostics, treatment, and even post-operative care. These are not just the buzzword, rather they represent a pillar of next generation healthcare system. And hence, this faculty development program aims to ignite the interdisciplinary discussion, foster research collaboration, and equip our faculty with insights into this evolving frontier. Now, so far, three topics which this faculty development program is going to focus. First is the imaging. We know that imaging starts from X-rays to MRI, the PET scans, and they are generally going in depth into the pathology of the disease. But now with the new development, it's going to the molecular level also. So it is, I think, in this point will be discussed during this FDP. So for the sensors as concerned, there are wearables and implantable sensors which have been used around the globe in a particular, and there is a new paradigm shifts into the continuous personalized healthcare to the from the hospitalized healthcare, starting from the glucose monitoring system to ECG patches to the smart stent and the neural sensors. We are moving from the hospital based and to the patient care, patient centric monitoring system at home. So what was once science fiction, such as a lab on a chip biosensor, is now a real laboratory reality. So for the biomaterials are concerned, biomaterials are no longer a passive. We are witnessing the emergence of the smart biomaterials that can respond to stimuli, deliver the drug, and promote tissue regeneration and wound healing. Hydrogels, nanocomposite, 3D printers, scaffolds, the potential application in orthopedics, cardiovascular cares, and the wound healing are the staggering. The development and use of these materials require a confluence of material sciences, physiology, ethical practices, and endurance of responsibility extend to guide the student in designing the biomaterials that are not only effective but also safe, affordable, and sustainable. So in the end, I must urge all of us to view the faculty development program not merely as a training module, but is a call for the action or call to the action, a call to reimagine the future of healthcare through the lens of innovation. Let us challenge ourselves to move from conventional teaching to transformational mentoring, from silos research to interdisciplinary program solving, and from passive learning to purpose-driven innovation. Thank you very much for patiently. Thank you.
And thank you, sir, for your kind words and illuminating us regarding the purpose of this FDP. It is now my great honor and privilege to invite our chief guest, a visionary academician and institutional leader who has made remarkable contributions to the field of engineering education and research in India. Professor R.S. Verma, the director of Motilal Nehru National Institute of Technology, Allahabad, is a highly respected figure in the academic community. Under his dynamic leadership, MNNIT has further strengthened its position as a hub of innovation, excellence, and impactful research. With a career spanning decades in technical education, Professor Verma is known for promoting interdisciplinary collaboration and fostering research ecosystems that benefit both academia and society. Sir, we are truly honored by your gracious presence at this faculty development program. I request you to kindly address our participants with your knowledge and wisdom. Thank you, sir.
Good morning, all of you. Can you hear me?
Good morning, sir. Yes, sir, we
good
can
morning,
hear
sir.
you,
Yes, sir.
Good morning to all of you, and thank you for inviting me for this very, very integrating topic and very timely topic of the faculty development program on smart health innovation, integrating imaging, sensor, and biomaterials. I congratulate all of the people MNIT faculty, department, Galgotia universities, and other people who are part of this. And I'm hopeful that this FDP program will help you to at least to give some boost or some kind of a intuition to do something different in this area. And this is the very prime area, and I've been working on these areas for the last 20 years, and I know how important it is. As said by the Professor Sharma also, that we are going to the molecular level of this to do the good health and find out the ways to do some of the technology which can help to design either the medicine or the other things, just like even the surgery also. And these are the tools which we are going to use it in this material, I think. Now, one of the things which I'd like to say to all the faculty, that you start thinking out of the box. Do not do the research or thought or to involve yourself in some of the things which has been done for the centuries or old technology that we want to improve this. You come up with some new ideas. With my experience, I can tell you, so far, none of the Indian companies make any materials, any sensing, any imaging devices which are world level. Even I can tell you dental things which are using the spatulas and all the materials is all imported right now. Even sutures, the people prefer outsides. Only one thing is developed, a heart valve, Chitra heart valve, which is the one of the thing which has been accepted all over the world. So I think we need to have a good people, good engineers, and good biotechnologists to think more of the things. Just like if you want to buy any instrument, we always think about either the German made or the American made or the Japanese made. Nobody talk about the Indian made. And our Prime Minister is also very much in this area, that we must do quality developed equipments, whether it's the imaging or sensor or any other things like that. Imaging is one of the things I think people are doing with AI and with the imaging things. Sensor also they are developing. But yes, biomaterial is one of the areas where you can think about how we can develop a material which can be used in these kind of devices. So think about the disease, think about the process, not only developing the drugs, but also think about the imaging and how to sense them. I think if you think about that, nowadays the blood glucose is just a sensor technology. You have to just prick something, one small tiny drop will tell you how much blood sugar is there. I think we have to have some kind of non-invasive methods to sense and find out the diseases. That is a challenge for us. We have not developed anything. So I think we are brainy people. We have technology, we know the science, we are hardworking people, but somehow we are not developing something, or we are not involving in the research area fulfilling. We are always worried about that I'm not getting the grants, so I cannot do it. Sir, you have to do it with find out the people who are working in your area and develop and send a project with a group of peoples. You will get it, because forget about individual research anymore. Until they do it in a group, you will not achieve anything. Think about the Nature and Science paper. There are 40 people are working, there are 10 labs are working together to find out some solutions. And that mentality has to be changed. I think private universities also has to play important role in this area also, because I know they have a little bit of infrastructure and also the people, but they can share with the institution of the government institution, tell them that, okay, let's develop a program, let's develop something projects and submit it together. I think that's why we are going to achieve it. So I again congratulate all the people who are participating or organizer here for having such a FDP program, and all the participants, please do it. Ask the question, ask the things to those who are giving the lectures. Find out some of the questions and they will answer you. Because most of the time what happened in this meeting, FDP people come, they give the lecture, nobody asks any question, nobody asks anything. I think we should have something. We should have some question answer session. We should have some, some kind of after faculty give them questions. Faculty should give the question what you have learned about that. So outcome is very important. Learning is very important. So thank you so much again, one more time, for inviting me. And I wish you all the success in this program. Thank you.
Thank you so much, sir. Thank you, Professor Verma, for your encouraging words and your esteemed presence. Sir has highlighted a very important point regarding the focusing on indigenous development of sensors, imaging, and non-invasive equipments. We really hope that with the shared knowledge, we will be able to move forward in this direction. Sir, we are really grateful to listen to your words of wisdom, and may we all take to heart what Sir has said today and move forward in that direction. Thank you so much, sir, for gracing this occasion. Moving forward, I would like to introduce, and it is my privilege to invite a stalwart in academic leadership, a visionary educator, and one of the major driving forces behind Galgotias University's journey towards excellence, our Honorable Vice Chancellor, Professor Dr. K. Mallikarjuna Babu. Professor Babu is a distinguished academician and administrator with a profound legacy in engineering education, innovation, and institutional development. With his extensive experience in research, teaching, and governance, he continues to shape the future of higher education with a focus on quality, interdisciplinarity, and global standards. Sir, your presence today not only inspires us, but reaffirms the university fostering a culture of innovation and collaborative academic rigor. We are deeply honored to have with us in this inaugural session of the Faculty Development Program. Sir, I respectfully invite you to deliver the address.
Good morning.
Good morning, sir.
Yeah. Now, we are part of this FDP on smart health innovation. So hosted by our School of Biosciences and Technology and Department of Biotechnology and Biosciences. So this is a wonderful to be here. And I, first of all, at the outset, I thank the chief guest for having accepted our invitation and being there and addressing the gathering. And I'm very amazing to see a lot of participants, 300 plus participants going on onto this FDP talks the credibility and quality of the FDPs that are being hosted and conducted by Galgotias University. Usually the online population, online connectivity will always be very number registered will be very high and participants will be very low. So this is the other way around, and where I'm indeed very happy to see that response. Having said that, any faculty development program, if you really look into the ethos, why this has been created, why this has been talked about loudly, that indicates it is the need of the hour. Need of the healthy lives and promoting well-being is being the SDG, and driving towards to that and bringing the systematic changes into it, bringing in all the innovations and required integrations, which is the need of the hour. So it's a rightly chosen topic for faculty development program. And I wish this by the end of this particular FDP, I think everybody should have a takeaway and everybody should have a kind of collaboration or network as far as possible. I always wish that to happen, as the chief guest also suggested, outcome. Without outcome, then probably we will not be able to really justify the activities we are doing, and not activities, the time, 300 people spending huge amount of time on a topic and without learning makes me really worried. So now rather we capture this learning in a different shift, then try to analyze this further continue. And coming on to smart health innovations are very much required in the context of country itself. So slowly from young nation to one day we'll also become aged nation, aged population, which is going to happen maybe 2 decades after. Let's say for that, we as a country need to be prepared. So such kind of programs really bring in the innovation in all the kinds of sensors and biomaterials. Biomaterials is one thing which is a tag, which is the need of the hour, and the kind of systems which we are talking about integrating. Maybe we have the help today of the technology. This technology is including artificial intelligence. We are trying to really work more on health-related sciences. Recently, our entire team, as a team, we have participated, right? We all went to gyms. So Government Institute of Medical Sciences here in Greater Noida, and we had a deliberation with all professionals, all doctors, right, medical community. So we have also had the opportunity to visit their labs, and our team is actively collaborating with these people, the GIMS faculty and Galgotia faculty start working together, at least bring some few innovations. That's what we are showing. So that falls under this particular topic today, FDP topic, Smart Health and Innovation. With a few thoughts of mine, and Galgotias University is a private university. Before I conclude, I just wanted to say what we are doing at Galgotias University. So Galgotias University is a private university established in 2011. So it's just the 14 years baby. So it start moving, inching forward and putting its clear imprints in the areas of teaching, learning, and then research and innovation, then move on to stepping towards the area of internationalization of education. All the NEP facets, we are there into it, but we have significant achievements, including SDGs. So now, having said that, our educational pedagogies are really changed a lot. So then we say, at Galgotias University, waging war on rote learning. The learning is the one which is causing the learning, the way the student learn. We would like to convert that from a rote learning to active learning. That means a passive to active. So now when we are talking about this, the kind of Gen Z and all the alpha generation which are going to come to the higher education have a different requirements, different needs of learning. So their practices are different. So now we are embracing all of them and move all the classrooms into completely digital mode, and AI is being driven into the classes. So all the classrooms are AI powered, and all our faculty have been trained by our mentor partner. So Nanyang Technological University through their Inspire cells. So all our faculty have been trained into how do we handle classes in a different mode, in an active learning mode. These classes really is attracting students to the classrooms. Hitherto earlier, that was not the scenario. So we used to get about 70%. Now it is 80, 90, 95% classes students are coming into. And this is their learning zeal is enhanced, and the learning style is also enhanced. So why very secret is we are matching their learning interests. So that's what the teaching learning side is concerned. In research side, we have been doing significantly well. As a young university, we are now around having a H-index of 90 plus, and then we are aim to go more than 120 in the coming year. That actually talks about what kind of focus we are moving on to the research, be it sciences, be it health sciences, or be it computer sciences, or be it law, be it humanities. We are there into research. And to give you one particular metric, which I would say that 1,000 research scholars are working in various problems into the societal. So all this multidisciplinary way, the research is also being driven. We do have an incubation center, very active incubation center. which is MSME host institution. And that way we also move, and so many of our alumni and our existing students have their own startups, and including I will also not shut to say that our faculty also have some startups. So this way, the research is also being nurtured. As a private university, we have to do a lot. And we do have some restrictions, but nothing stops Galgotia University. No blocks, right? We keep on aim only for progress. We move on to the progress. So that way, I say that such kind of faculty development programs, we also do rigorously learn out of each and every activity our faculty does, and then we encourage them. So I also invite all the participants here who are from other institutions. So visit us, Galgotias University, right? Visit us any point, whenever you are here. So please do make a point to visit us, collaborate with us and our faculty. Then let's work together to build a greater and stronger India that is envisioned by Honorable Prime Minister Modi, right? So to build Atmanirbhar Bharat, so we all have to join together, and together we will be able to succeed in our endeavors. With that few thoughts of mine, I wish all the very best to this faculty development program on health, right? So wish you all the very best, and thank you. Jai Hind, Jai Bharat.
Thank you for your kind words and your inspiring speech and unwavering support for academic excellence, as always. Sir has always been our pillar of strength and has always shown his full support towards innovation, research, and scientific excellence. We are really fortunate to hear from you on today's occasion, and we hope that you will be a part of our further ventures and you will continue to support us throughout our events and all the programs. Thank you, sir. We are really grateful to have you here with us today. Thank you.
Thank you, Sumedha.
Thank you, sir. Thank you so much. So, to formally conclude the inaugural session, I would now like to call upon and invite Dr. Saurabh Bhardwaj, the Faculty Development Program Coordinator from MNNIT Allahabad, to deliver the vote of thanks.
Thank you, Dr. Mukherjee. I hope I am audible.
Yes, you are audible, sir.
Respected dignitaries, esteemed speakers, honorable guests, and my dear participants, it is my honor and privilege to deliver the vote of thanks on behalf of the Faculty Development Program Coordinating Committee for this impactful and promising inauguration of our FDP on smart health innovation, integrating imaging, sensors, and biomaterials. First and foremost, I express my deepest gratitude to our chief guest and chief patron, Honorable Professor R.S. Verma sir, Director MNNIT Allahabad, for his valuable presence and motivating address, which truly elevated the tone of this program. I also extend heartfelt thanks to Honorable Chancellor of Galgotia University, Mr. Sunil Galgotia, and Dr. Dhruv Galgotia, CEO of Galgotia University, for their visionary leadership and continued encouragement in advancing academic and research collaborations. A special note of appreciation to Professor Dr. K. Mallikarjuna Babu, Honorable Vice Chancellor of Galgotia University, for gracing today's inaugural session and inspiring us with his words of wisdom and academic foresight. I sincerely thank to Dr. Ajay Bharti, Head Officiating, Department of Applied Mechanics, MNNIT Allahabad, for accepting our invitation and addressing the participants. My sincere appreciation goes to all the dignitaries who shared their insight this morning. Dr. Uvanesh K, FDP Convener and Assistant Professor, MNNIT Allahabad, Dr. Gaurav Kumar, FDP Organizing Secretary and HOD, Department of Biotechnology and Bioengineering, Galgotia University, Professor Abhimanyu Kumar Jha, Dean, School of Biosciences and Technology, Galgotia University, and FDP Co-Convener. I also extend my greatest heartfelt thanks to Professor R.S. Sharma, Distinguished Professor, Galgotia University. Thank you for your inspiring contribution and academic stewardship. My sincere appreciation extends to all the session coordinators of this FDP program, Dr. Sumedha Mukherjee, Dr. Dheeraj Ahuja, Dr. Somnath Roy Chaudhary, Dr. Soumyadipta Rakshit, Dr. Anjali Priya, whose meticulous coordination and support will be instrumental in ensuring the smooth conduct and success of each technical session throughout this FDP. A heartfelt thanks to the organizing team from both the universities, Galgotias University and MNNIT Allahabad, whose meticulous planning and coordination have brought this FDP to life. To our 2 keynote speakers and 25 invited speakers, we are truly grateful for your willingness to share your expertise over the coming week. We are confident that your sessions will be intellectually enriching and practically meaningful. Last but not the least, I thank all the participants from across India for joining us and making this a truly national academic gathering. Your enthusiasm is the driving force behind the success of this program. With these words, I once again thank each and every contributor for their role in making today's inauguration session a resounding success. Let us now proceed forward with shared purpose and academic spirit. Thank you. Jai Hind.
Thank you, sir, for your kind words, and thank you for addressing the gathering today. And thank you. So, and also we thank Dr. Bhardwaj for acknowledging all the efforts of all the stakeholders with such grace. Now we have come to the conclusion of our inaugural program, and now we'll be moving towards our first technical session of the day. So let us hope that this will be a week of learning, celebration, and we will celebrate new knowledges about smart healthcare, and we will celebrate innovation, and we will have with us the wisdom. So to celebrate wisdom and innovation and knowledge, I would like to start our technical session with invoking the goddess of knowledge, Devi Saraswati, with a Saraswati Vandana. After that, we'll proceed towards our first technical session. Thank you. Thank you all for being with us and paying tribute to Devi Saraswati, the goddess of knowledge. And now we'll move towards our first technical session. And we'll begin our first technical session of the FDP with the first esteemed speaker of the day, Professor R.P. Tiwari from MNNIT Allahabad. It is an absolute honor to invite him, who is a renowned academician and a researcher in the field of robotics, biomechanics, and biomedical engineering. He completed his Bachelor of Engineering in 1990 from MMM Engineering College, Gorakhpur, MTech and PhD in 1993 and 1999, respectively from the prestigious Institute of Technology at BHU Varanasi. He has around 25 years of teaching and research experience and started his career as a scientific officer at IMS BHU and joined as a faculty in the Division of Instrumentation and Control, NSIT New Delhi. Later, he moved on to MNNIT as a faculty at the Department of Applied Mechanics in 2005 and is currently working as a professor. He has held several administrative positions like head of the Department Applied Mechanics Department from 2015 to 2017, member of Central Purchase Committee, member of the Board of Studies, member of Academic Council, chairman at Library, and others at MNNIT Allahabad. He has served as Dean Planning and Development and part-time Chief Vigilance Officer at MNNIT Allahabad. He was on deputation as Central Project Advisor MNNIT scheme of Ministry of Education, Government of India. He has published more than 100 papers in high-impact international journals and conferences of repute. Dr. R.P. Tiwari has worked closely with the government as well as the industry on various problems and has successfully laid and completed many projects and programs at the national and international levels. Dr. R.P. Tiwari has carried out funded projects worth more than 1 crore and consultancy to various state government organizations, central government organizations, and public as well as private sectors worth more than 30 lakhs. We are privileged to have him today with us, and he will be enlightening us on the topic Innovation in Healthcare and Engineering. Sir, we warmly welcome you and request you to kindly begin your session. The stage is all yours.
Hello. Good morning to all of you. Am I audible?
Yes, sir, you are audible.
So, good morning to all of you. The topic of my presentation is smart healthcare innovations and startup ecosystem for students in smart healthcare. Thanks for nice introduction, Dr. Sumedha.
Thank you, sir. We are really grateful to have you with us today. It is a prestigious occasion to hear from you.
Actually, slide is not moving. That's why. Okay. I will not keep it in full screen. So what is a smart healthcare? I will just give you a brief idea and what is the startup ecosystem for students. We all faculty members are here for this FDP. So I would request all of you to please motivate our students to do innovation and research in healthcare. So smart healthcare is a technology-driven model of medical care that leverages data connectivity, automation, and real-time monitoring to deliver proactive, predictive, and personalized healthcare solutions. So key drivers of smart care: artificial intelligence and machine learning, Internet of Medical Things, big data and cloud computing, advanced medical image technologies, smart biomaterials, wearable mobile health applications, personalized medicine, telemedicine, and remote care. Now, key drivers of smart health. First one is the artificial intelligence and machine learning. A lot of work is going on in this emerging area. When we are talking about the smart health, it enables predictive diagnostics, image segmentation, and pattern recognition. It is mainly used in radiology, pathology, and patient outcome prediction. You can see the health monitoring and wearables. The block diagram of this one is where we can use this AI and ML technique in healthcare, while virtual nursing assistance, cyber security, gene editing, AI, robot assisted surgery, and various other applications like personalized healthcare plans in selecting personalized healthcare plans, cancer research, dose error reduction, medical diagnostic, drug development, and rare disease diagnostic, early detection of rare disease for diagnosis and treatment purpose. So this is the key drive. First one is the artificial intelligence and machine learning. When we talk about the smart healthcare innovation, a lot of work is going on and a lot of work is still required in this emerging area when we talk about healthcare sector. This is the basic block diagram how AI works. You might be knowing this. Data collection, data pre-processing, model selection, training of the model, testing and evaluation, model optimization, deployment, continuous learning, and finally we will get our answer or the solution. So when we talk about AI in healthcare involves uses of machine learning, natural language processing, computer vision, and other AI technologies to process and analyze health data. The aim is to extract meaningful insights, support decision making, and automate or augment tasks traditionally performed by the healthcare professionals. So this is the machine learning, image recognition, artificial intelligence, and natural language processing. And deep learning is a part of machine learning. So when we talk about machine learning, this is a core component of artificial intelligence in healthcare. ML algorithms learns from vast amount of health data to identify patterns and make the predictions. For example, they can predict disease progression, treatment outcomes, or like a hospital admission, etc. Second important thing is the natural language processing, that is NLP. NLP is basically enables the computer to understand and interpret human language. In healthcare, NLP is used to extract valuable information from unstructured data such like clinical notes by the doctors, research papers, and patient feedback. You can see this. NLP is basically combination of audio, video, and text messages. Entity extraction, sentiment analysis, it is included. NLP includes all these things: raw language processing, topic classification, relationship extraction. So this is also an important feature for healthcare innovation. A lot of work is required to be done in this area also. Computer vision. Next is the computer vision. This aspect of AI involves enabling computers to interpret and analyze visual data. In healthcare, computer vision is applied. Am I audible now?
Yes, sir.
Yes. In healthcare, computer vision is applied in medical image analysis, such as identifying tumors or any other abnormalities in MRI or CT scan or PET scan or X-rays. Now, what are the various applications of computer vision in healthcare? Tumor detection, intelligent medical training, hospital hygiene inspection, disease and infection prevention, patient identification, cancer detection, vital organ health monitoring, detecting tissue structural changes, etc., etc. Next thing which is important is the predictive analytics or predictive analysis of disease or any abnormality. AI algorithms can analyze diverse data set to predict future events or outcomes. You can say it early detection of any disease or any abnormality in human subjects. In healthcare, this can mean predicting disease outbreaks, identifying patients at risk of developing certain conditions, or forecasting the demand for the health services. So this is basically artificial intelligence, modeling, data mining, machine learning, and statistics. It combines for the future prediction, or we can go for the predictive analytics based on these parameters. Now, personalized medicine. AI supports personalized medicines also by analyzing genetic, environmental, and lifestyle factors to tailor treatment to individual patients. This can lead to more effective therapies with few side effects. Robot process automation, it is also an emerging area of research in healthcare innovation. RPA involves the use of software robots to automate repetitive and routine tasks in healthcare administration, such as data entry, appointment scheduling, billing, and other things. This is also the Internet of Medical Things is also an emerging area in healthcare innovation. It is basically Internet of Medical Things, network of connected medical devices that collects and transmit data, enables remote monitoring and personalized healthcare. Smart watches, examples of this Internet of Medical Things. You can connect several medical devices together and you can collect data. Examples of this is smart watches, glucose monitoring, and implantable devices. This big data and cloud computing is also an emerging area of innovation and research in healthcare. It allows large scale, because health data is a very big voluminous data, you can say, allow large scale storage, integration, and analysis of patient data, facilitates real-time decision making and telemedicine, supports electronic health record. You can see, then when we talk about electronic health record, it is a cloud-based data. We have to keep the data on clouds. So when we talk about telemedicine application models, we can go for tele-surveillance, tele-diagnosis and tele-treatment, tele-monitoring, tele-education and teleconferencing, tele-surgery or robotic surgery, tele-radiology, and several applications of telemedicine are there. These are the emerging areas of research and innovation when we talk about the healthcare innovation and research. Advanced imaging technologies, CT, MRI, PET, and ultrasound now enhanced with the artificial intelligence. Improve accuracy, speed, and automation of diagnosis, important in oncology, cardiology, and neurology. So with the help of AI, we can improve the performance, accuracy, speed, and automatic detection of the diseases with the help of AI and ML techniques. Smart biomaterials responsive to the biological or environmental and biological parameters, you can say, responsive to pH, temperature, or electrical stimuli, used in tissue regeneration, drug delivery, and implant design and development. These smart biomaterials must be biocompatible, and it is not necessary it is biodegradable, but nowadays biodegradable biomaterials are popular because a lot of, as some of the speakers were telling about the degradable sutures and other things. So nowadays biodegradable materials are becoming popular day by day. So they are replacing the biocompatible non-degradable biomaterials. So we are using the smart biomaterials in tissue engineering, wound healing, sensor, drug delivery, and degradable implant and non-degradable implant applications also. So robotics and automation is also an emerging area of research and innovation when we talk about the healthcare. Nowadays, robotic assisted healthcare surgery for precision is important and popular nowadays. Automation in diagnostics and hospital workflows, reduce human error and increase efficiency, minimizes contact in infection cases. When we use robotic devices or robotic nursing system, then it minimizes the chance of infection to the doctor or the nurses. Wearable and mobile health apps, continuous health tracking, encourage preventive care and early detection, empower patient to monitor their own health. Telemedicine and remote care, doctor-patient interaction via video or app-based platforms is becoming popular day by day. Increased reach in rural and underprivileged areas, save time and cost, especially for chronic disease management. When we talk about the personalized medicine, tailored treatment based on genetic, biomarker, and lifestyle data, it is powered by AI and molecular diagnostic techniques. Now, this is the different aspects of smart healthcare when we are comparing smart healthcare with traditional healthcare. You can see the difference between these two. When we talk about the approach, the traditional healthcare is the reactive, means treatment after disease onset, but while the smart healthcare is the proactive and preventive, means real-time monitoring and prediction of the disease and early detection and diagnosis and treatment of the disease. Access. When we talk about the traditional healthcare, it is hospital or clinic dependent. When we talk about the smart healthcare, it is remote and decentralized, telemedicine, mobile app, etc. Technology used, conventional tools are used when we talk about the traditional healthcare. When we talk about the smart healthcare, AI, IoT, big data, as I have told you, wearable devices, robotic devices, smart biomaterials, etc. Data handling, normal paperwork or EHR. In the case of smart healthcare, real-time data analytics and cloud integration. When we talk about the personalization, generalized treatment plans for individual patients. When we talk about the smart healthcare, personalized and precision medicine based on data. Cost efficiency. This traditional healthcare is costly because we need to go for hospitals and for various tests. When we talk about the smart healthcare, it is optimized via remote diagnostics, early detection, etc. Response time is delayed. In case of traditional healthcare, depends on the physical visit. When we talk about the smart healthcare, instant alert and interventions are possible. Follow-up and monitoring, manual and intermittent when in case of traditional healthcare, but in case of smart healthcare, it is continuous and fully automated. Focus, when we talk about the traditional healthcare, it is a disease-centric. When we talk about the smart healthcare, it is wellness-centric. So smart healthcare, that's why smart healthcare is becoming popular day by day. So India's healthcare challenges, what are the real challenges in India? You need to work in this rural and urban divide. Around 65% of the population lives in rural areas with limited healthcare access. Low doctor to patient ratio, 1:1500. High output of pocket expenditure, over 60% of medical expenses are paid directly by the patients. Infrastructure gaps, shortage of hospitals, ICU beds, imaging facilities in tier 2 and tier 3 cities. It is a very big challenge for tier 2 and tier 3 cities to meet out these infrastructural gaps. Skilled manpower deficit, lack of trained technician, nurses, biomedical engineers in tier 2 and tier 3 cities. Lack of early diagnosis, late detection of diseases like cancer, diabetes, and TB, etc. These are the major healthcare challenges in India. Now, what are the opportunities for innovation in healthcare? There is a need to develop affordable point of care devices, low cost diagnostics and mobile labs for rural areas, AI powered telemedicine platforms, expand access to quality consultation, wearable device and internet of medical things for continuous and vital monitoring for chronic care, smart imaging and teleradiology, AI based interpretation for faster diagnosis, digital health records, centralized accessible health data infrastructure, etc. I am mainly focusing on why we are focusing for the students, because students are the backbone of innovation. Why students should care? High career potential, your opportunity to innovate and impact. High care potential, rapidly growing health tech sectors like AI, IoT, bioengineering, bioinformatics. Demand for interdisciplinary skills means coding skills, biology, knowledge of biology, hardware knowledge, designing knowledge. All these, basically this healthcare innovation is an interdisciplinary research or innovation area. Global opportunities in research labs, startups, hospitals, and medtech companies. What is the role of students when they are working in healthcare innovation and research? They can get the role of biomedical data scientist, AI ML engineer in healthcare, regulatory and clinical validation analyst, bio entrepreneurs, and startup founders. Innovation playground, real world unsolved problems in healthcare need local solutions. Government and institution support, hackathon, funding, incubation, a lot of government schemes are there. Some of the successful stories of students who worked in the healthcare, access to free tools, Python, Raspberry Pi, TensorFlow, 3D printing, etc. Societal impact, improve rural access, reduce diagnostic delay, lower the cost. This is the major challenge for Indian population. Work on meaningful projects with real outcome, public health, maternal care, disability aids. There is a need to work in these areas with some innovative solution of the real challenges. A smart health innovation can save the lives and change communities. A student can be the change makers, startups for Bharat, not just for profit. This is they can change the scene when we talk about the healthcare innovation, because most of the healthcare devices we are importing. we are exporting from outside. So when we talk about the rehabilitation devices or assistive devices, most of the devices we are importing it from other countries. And it is very costly. There is a need to make these devices. Make in India. Our Prime Minister is also urging to make the medical devices in India, and he is promoting a lot of government initiatives for this purpose. So next thing is the innovation ecosystem. When we talk about the innovation ecosystem, college, university, industry, non-government organization, government organization, investors and funders should come together and work together to develop this innovation ecosystem in India. There is a need, urgent need of this thing. The government is promoting through various schemes, but there is a need that colleges, universities, industries, NGOs, government organization, and investors and funders, they should come forward and work together to solve the real life challenges in healthcare. What is innovation in healthcare? Healthcare innovation is the process of developing and applying new ideas, technologies, methods, or models to improve health outcomes, reduce costs, as I have told you, to make the products in India, improve health outcomes, reduce costs, enhance patient experience, increase accessibility. So when we talk about the innovation in healthcare, there are different categories. Product innovation. Product innovation means development of a new tool, device, or a drug. Examples are smart inhalers, wearable devices, wearable ECG monitors, bioengineered skins, etc., etc. When we talk about the process innovation, there is an improvement in how healthcare services are delivered. There is a need to improve the healthcare process. Third is the business model, new way of organizing, financing, or delivering a patient care, subscription-based diagnostic, teleconsultation, and a lot of other things. Fourth type is system innovation. System innovation means a structural change at a macro level, policy integration and regulation, like government schemes. You can say Ayushman Bharat for healthcare, NDHM, that is National Digital Health Mission, etc., etc. Key features of true healthcare innovation, we can say, solves a real clinical or operational challenges. We should, all the technical institutions and medical colleges should work together and work to try to solve a real clinical or operational challenges that doctors or physicians or surgeons are facing. Is scalable and cost, the solution must be scalable and cost effective. It must be clinically validated. Regulatory compliant is very important when we talk about the regulations of medical devices in India. 2-3 years back, there was no regulatory compliant, but now there is a Medical Device Regulation Act that is already approved by Government of India. Patient and provider friendly, it should be patient and provider friendly. These are the things that is required to be taken care while going for the healthcare innovations. So when we talk about the students, we should urge our students to come forward for healthcare innovations. Why it matters for students? Identifying inefficiency, proposing, students can go for proposing a tech-based solutions for the real clinical or operational challenges. Innovating does not need a degree. It needs insight and empathy. This is the technology readiness level. You might be knowing this, TRL1 to TRL9. Technology readiness level are a scale used to assess the maturity of a technology during its development from conceptualization to real world applications. Basic principles observed, that is TRL1, concept formulation TRL2, experimental proof of the concept TRL3, lab validation, where most of the labs are doing this lab validation. We are going up to TRL4 level. So relevant environmental testing, that is TRL5, prototype demonstration TRL6, system prototype in clinical use, TRL 7, then full system qualified, that is TRL 8, and actual system with proven and all the regulatory certification, that is TRL 9. So there is a need to develop technology readiness level up to TRL 9. We can take help after TRL 6. We can take help from or collaborate with the industries and the R&D organization to give the product in market. So this is the healthcare. I will just conclude within 5 minutes. So healthcare innovation pipelines. First is the ideation, identifying an unmet healthcare need and conceptualizing the technology-driven solution. Prototype, building a functional model that is hardware-based, software-based, or any combination of biomaterials or any other assistive device. Validation, testing for the technical feasibility and clinical relevance. IP filing, protecting your idea with a patent or copyright or design, ensuring compliance with the standards, regulatory authorities, that is CD, SCO, ISO, FDA, depending on the product. And then we can go for the market entry, deploying through pilot, partnership, and or commercializing the check. There are different national schemes you might be knowing. Smart India Hackathon. I will just go through quickly all these slides. These are the national innovation initiatives. Government of India is promoting a lot of schemes for promoting a startup ecosystem in India. Yukti, that was the Young Indian Combating COVID with Knowledge, Technology and Innovation. This is developed by the Ministry of Education Innovation Cell, IIC. Ministry of Education Innovation Cell, that is IIC. Other notable program, Atal Innovation Mission, National Innovation Foundation, Startup India, Startup UP. There are several schemes launched by the Government of India as well as Uttar Pradesh government. You can take full benefit of all these schemes. So I will not go into the relevance to students in smart healthcare. You can see these slides. BIREC is also promoting a lot DST schemes for seed funding, patent filing, prototype support. A lot of government schemes are there. BIREC and biotech startups are also supported by Department of Biotechnology. Major funding schemes of BIREC, you can see these. This is important for students and innovators. All these details are available on Government of India website, Startup India. Now, one thing I would like to tell you, we are also having an incubation center here at MNNIT Allahabad, Innovation and Incubation Hub Foundation, MNNIT. We are mainly promoting the startups in healthcare and Internet of Medical Things. We are also requesting you all to please visit to our labs. We are having this Internet of Medical Things. For that, we have a 5G, Government of India 5G lab is also available. You can do your innovation and research in collaboration with our incubation center in 5G technology. So what is an incubator? Just I will quickly finish this. An incubator is a supporting environment that helps early stage startups, including student-led one, offering workspace, mentorship, funding access, technical resources, and networking opportunities. So we are providing following kind of support for prototype infrastructure, mentoring support, business support, funding access, IP and legal help, and networking. So I would also request Galgotias University that we can sign an MOU with MNNIT as well as with our incubation center in near future for developing co-incubation culture at both the institutions and university. So I would request Galgotias University, we will quickly sign an MOU with MNNIT as well as with our incubation center for developing this, promoting this startup ecosystem in Uttar Pradesh. I will not go, this all you might be knowing. thank you all. That's all from my side.
Thank you so much, sir.
Thank you, sir, for your very enlightening words and for your engaging and thought-provoking session. Your insights on innovation in healthcare engineering truly set the foundation for the discussions we aim to pursue in this program for the current week. As a token of our respect and appreciation, we would now like to symbolically present you with a virtual memento. I request Dr. Gaurav Kumar to kindly present a memento to Dr. R.P. Tiwari.
Respected sir. Hello. Respected sir. Ravi sir, kindly accept this e-memento
Yes, yes.
as a token of gratitude from the Galgotias University. And sir, we are fortunate that you have invited me and providing me the opportunity to be collaborate with you in your department to organize this FDP. And sir, we will certainly sign a MOU with the incubation center. Thank you so much, sir.
Okay, Dr. Gaurav.
Participant, if you have any query, you can unmute yourself and ask your query if you have any.
The session is open for questions and answer. You can ask Professor R.P. Tiwari sir if you have anything to ask.
Okay, thank you. Thank you, Dr. Gaurav and Dr. Uvanesh. Thanks for giving me this opportunity.
Thank you.
Okay, sir. Okay, sir. Thanks for your time, sir.
डाइनिंग के पास
Thank you, sir.
Gaurav sir, shall we invite Dr. Chandan Kumar for this next session?
Moving forward, I am pleased to invite our next distinguished speaker, Dr. Chandan Kumar from Supol College of Engineering, Bihar. Dr. Chandan Kumar is working as an assistant professor at ECE department at Supol College of Engineering, India. Additionally, he is also the Dean Academics and Head of Research and Consultancy in the same college. Before his current position, he worked as JSPS Fellow at the Nara Institute of Science and Technology, Japan. He also worked as Institute Postdoctoral Fellow at IIT Bombay, India, and as an Assistant Professor at the Department of Electronics and Communication Engineering at MNNIT Institute of Technology at MNNIT, India. He received a bachelor's degree from Muzaffarpur Institute of Technology at Muzaffarpur and from the department and a master's degree from NIT Durgapur in 2011 and 2014 respectively. He obtained his PhD from the Department of Electronics Engineering, Indian Institute of Technology, BHU, Varanasi, India in June 2019, and he is also a recipient of the DST Cefipra Fellowship 2018 and IC Impacts Fellowship 2016. His current research includes flexible electronics and thin film hybrid devices for solar cells, photodetectors, and sensing application. Today, he will be guiding us through this topic of organic thin film devices for biochemical sensing applications. Sir, the floor is all yours, and we look forward to listening to you.
Thank you, Dr. Mukherjee. Thank you, Dr. Mukherjee, for nice introduction. I am audible.
Chandan sir, your voice is not audible.
Hello, Gaurav sir, can you hear me?
Yes, sir. Yes, sir. You are audible now.
Sir, you are audible.
Thank you.
Thank you, Dr. Mukherjee, for a nice introduction. So, slide is visible.
No, sir, not yet. Now it is visible, sir.
Thank you, Dr. Mukherjee, for a nice introduction. So, today I will give a brief introduction about the organic devices for biochemical sensing of human or like a chemical wastage. So, like a living cells, metal ions, or so in this, I will go one by one. First, I will share. Uh,
Sir, your voice is lagging, so you can switch off your camera.
Yeah.
Yes. Okay.
So this is some research summary of still lacking problem. Gaurav sir.
Yes, sir. Yes, sir. You can continue,
Sir,
sir.
you can talk. Actually, we need to mute participants.
Yeah.
Okay, thank you.
So these are some summary of my research. So I will go now to the my outlines. So first we will introduce about the evolution of electron. So first of electronic devices. Now we are talking nowadays like a
Chandan sir, your network is it is like very it is not stable actually.
is not.
Sir, it is not stable actually, your network. We were not able to listen you. Sorry, we will be coming back in few minutes. It seems some network issue.
बहुत धूप आती है वहाँ भी, यहाँ बैठ नहीं सकते, इतनी धूप आती है यहाँ, तो हम ब्लाइंड से लगे तो
We request participants.
Sorry, we have issue with some internet. So Dr. Uvanesh will share PPT. I will talk on that. I've already shared with Dr. Uvanesh, so he will share PPT.
Yes, sir. We
Yes,
already.
yes, yes.
Sorry.
Yes, I'm working on. Give me a few.
That is why I'm asking participants to keep a little bit of patience. So we'll be back online soon with his resourceful slides for today. And we request participants to be a little bit patient, and we'll be soon back with the session. Thank you.
So is it okay?
Yeah, it's visual. Go to, I think, fifth or sixth slide.
Which slide, sir?
Fifth slide, when we topics. Sure,
Okay.
yes. So first we will start with the thin film devices. Then we will go for the biosensing application as well as gas sensing applications. So next slide we have introduction of thin film. Next slide. So we all know the electronics has started with the introduction of transistor. So first transistor was developed in the Bell laboratory. So after that, we dream about different electronic devices nowadays, like handheld devices or like a portable devices, all are feasible with the development of transistor as well as ICs. So in next slide, we will show the development of electronic devices. So concept of point contact devices were first formulated by Baron, but first experimental demonstration was done by Jesse Vosch in 1901. So in this, they have developed the rectifying behavior of semiconductor devices, then the development of FETs, MOSFETs, and first thin film devices were developed in 1961. And then on we have go for the polymers and organic defects, and now the in 2009 first FTM based OFET was developed, and by us we have developed the first PQT based FTM devices for the uh, phototransistor as well as sensing applications. Next slide. So these are some applications of organic devices like in chemical sensors, light emitting devices. So beauty of these devices is that these devices can easily implanted into humans. So because these are highly biocompatible, non-toxic, and with time to time they can degrade in 1 year, 2 years. Some chemicals take up to 10 years. So these are flexible and implantable for the health applications as well as other medical applications. Next slides. So these are some example of flexible devices like e-papers or like RFID tag. Their organic devices can be used. So now next slides. We have the CMOS integration, everyone know. So conventionally silicon-based devices are used for the integration of CMOS. Nowadays we are in the phases that we can integrate P-type and N-type thin-film devices to form CMOS devices, and that will further will synthesize or like a motivate us to develop ICs-based organic devices. The advantage of CMOS generally include their low power consumptions and reliable and robust. These advantages can also be used for the organic devices. Next slide. So these are the example of how we grow from device to system. So first we make a device like a transistors, then we make a circuits, then we form a gate, and then using gates we make models, and different models make our complete system. So integration of high performance system is made like this. Now we will move forward to the thin film devices. Next slide. So thin film devices, we start with non-conventional substrates. So conventional substrates are normally silicon or other SOIU. So in non-conventional, we use thin substrates of like a glass or like a plastics. These substrates are used for the development of thin film devices. Then we deposit a thin film using non-conventional deposition methods like a spin coating, like a floating transfer method, a spray analysis, or these different methods can be used. And then we finally deposit other layer, and at last we develop the metal contacts. Next slides. So these are some characteristic of organic electronics. So these organic are low cost, flexible, process simplicity, and low temperature processing. It's like a temperature processing. Normally, silicon goes up to 600 degrees C or higher, and for hydrogen silicon, all amorphous silicon also 300 degrees C. But organic semiconductors, we developed it around 100 degrees C, and its carrier mobility goes beyond 1 centimeter square per volt second. Nowadays, up to 10 researchers are reporting. Next slides. Normally, we used colloidal inks, semi-transparent semiconductors, CNT, semiconductor insulating other materials, nanoparticles, quantum dots, and some adhesion promoter. These are some material used for the development of thin film devices. Next slides. Two most common technique generally go for the development of thin film devices, especially for organic, is spin coating and unidirectional floating film transfer method. The unidirectional floating film transfer method had added advantage over the spin coating. Normally, spin coating is used to make devices on a thin film, but their orientation is not a uniform. They are like a force driven formation of film, so their conductivity is relatively low. On the other hand, when we deposit like a thin film on a film using FTM, then their direction is uniform. So these give higher conductivity as compared to spin coating. Other methodology we generally use like a thermal evaporation, electron beam. These are for the deposition of metals in some cases. Next slide. These are some example of how we use organic fits for making driver circuits. So in this, we replace a conventional transistor with organic transistors. We provide some capacitor and resistors to form a driver circuit. The requirement of these driver circuit is the low voltage operation. These devices must be operated at low power, and we know that organic transistor consume very less current, so the power low voltage operation possible with these devices. Its mobility nowadays increased beyond 10 centimeter square per volt second, so it is most suitable for these applications. Next slide. So other example of logic circuits. So logic circuits can be made using p-type organic devices and n-type organic transistors. So we can make CMOS also using organic devices. At the same time, some organic materials show ambipolar characteristic. So in this we mix both P and N type characteristic. So these two devices based on applied voltage at gate can be used as a ambipolar. So either P type or N type it can be used. Next slide. These are some device structures. We can fabricate all these structures using organic transistors, organic devices. So like a photo conductor, Schottky photodiode, or PN photodiode, MIS diode. All diodes or transistor can be made using organic thin film devices. Next slides. So these are the operation of two terminal devices. So we applied positive and negative on two terminal devices, and their operation is similar to the conventional devices like a LDR or maybe PN junction diode using silicon. We make similar devices using organic devices, organic materials. Next slide. So these are four structures for three terminal devices. Organic transistors are made in four structures. So among these four structures, top contact, bottom gate are more favorable due to its fabrication strategy as well as highest uh, highest field effect mobility in these structures. Next slide. So for operation, we applied gate voltage at gate terminal, and with that, the HOMO level and LUMO level is changed or like inclined, and the carrier transportation increase. So these are the operation of thin film transistor. Next slide. So challenges in the thin film transistor. So some challenges in thin film devices is high mobility, low voltage operation. Approx 10 years ago, it is very challenging that high mobility is difficult to achieve in organic material based transistor. But nowadays we can see that more than 20 centimeters square per volt second achieved, and operation of that transistor all are also below 1 volt. So when we use different gate dielectric material, we change this operation, and one of challenges reduced. Next is contact resistance. Nowadays contact resistance is also reduced, and environmental stability is nowadays most of organic materials are coming with this more environmental stable. So these are more suitable for the futuristic devices for flexible purpose. Next slide. So in next slides, we will discuss some applications as a biosensing, heavy metal ions, and gas sensors. So normally biochemicals are present in the living cells, heavy metals or gases. These are some biochemicals that are living inside our organic foods, or maybe like organic waste. So we are focused on developing biosensor for detecting living cells, heavy metals, and gases. Biosensor also monitor healthy environments, and ammonia is the most common extracted gas from biowaste. Next slide. So first we will discuss biosensing applications and then gas sensing applications. Next. So in this we have developed uh, uh, devices for detection of cell growth on the surface using electronic setup. So nowadays electronic setup are developed for the detecting of growth of cells. These are when the cell growth, the conductivity on the surface is changed. So this change in conductivity is measured using electronic devices. So our focus is a develop electronic device, and over that we implant the cells that change the conductivity of that semiconductor materials, and this semiconductor will be organic, so that their change in so that the organic film the cell will interact with organic semiconductor faster, and their change we can measure effectively. In conventional semiconductor like a silicon, cell interaction is very slow, and therefore their detection is very slow. Next slide. So in this, we develop like a thin film of uh, zinc oxide, and over that we develop cells, so their interaction with the cells is relatively lower. When we further increase with like a organic substance, like when we do not treat gerano more, because when we anneal gerano at higher temperature, all the organic parts available in the zinc oxide uh, compound will eliminate. So when we annealing at lower temperature, we found that the sensing mechanism relatively higher as compared to anneal at higher temperature. So in this, we see that with the time, the cell has growth, and after growing, the conductivity is increased. Next slide. So in this application, we use MOS2 and onion derived carbon quantum dot for mercury detection. So in that we started with the organic byproduct of onion, and we synthesize carbon quantum dot. Over that we disperse MOS2 flags, and that is showing that uh, high sensitive towards the mercury ions. These mercury ions are normally present in waters, or especially in like sea waters from where fish are coming. So nowadays, lots of disease found in fish due to the excess presence of lead, excess presence of mercury. So this mercury should be detected before eating this grade of fish. So this type of handheld devices was developed during my postdoc time. So this is in trial phase still, so that commercialization can be possible. So in this, we put a small drop of water containing mercury over the strips of for these handheld devices. We see that a significant change in the conductivity is shown, which is converted to the detection level, and this can be go beyond the detection level up to ppb levels. Next slide. Other applications of functionalized organic functionalized carbon quantum dot is that they can also leading to detection of lead. So lead are also, they are showing significant change in the conductivity when interacted with the lead. So this lab level setup have developed for the sensing of lead, and they are showing the significant uh, change in their conductivity. Next slide. So in this, we have developed a functionalized security for arsenic sensor also. In this, we see that these devices, when functionalized with a different sulfur group in this organic, then the handheld devices is sensitive to the arsenic detection. We have also developed handheld devices for this purpose. Next. So now we go for the gas sensing applications. Next slide. So gas sensors are nowadays very common. These are used for the monitoring of environmental gases, as well as the organic wastes coming from homes, or maybe like industry wastes. So these sensors based on either chemical absorption or physioabsorption. So most commonly, gas sensors show sensitivity, selectivity, detectivity, their response time and recovery time, and their stability and lifetime. Next slide. So this is the sensing process of a gas sensor. Normal human nose, smell, brain stimulate, and neurons performs, and finally results coming. So similar way we developed electronics devices that their purpose is acquisitions of data from gases. Then they process data in form of electrical signals, then comparison of data by neural system, and finally results came. So this is the most common process of thin film devices based gas sensor. Next slide. So these are non-conventional devices developed using organic polymers. So this is developed on polyimide substrate that is flexible. Over that we deposited PQT organic polymer, and then we deposited AU electrodes, and you can see that this flexible nature of these devices, that we can strip this on a human body or anywhere, because this is very flexible, and the performance do not degrade over the like when we bend, or like we can hold in pockets, or like is keep anywhere. Next slide. So in this, we can see that interaction with the ammonia gas with the PQT molecules. So when interact with the PQT molecules, there is change in response. We can see significant response with the interaction with the ammonia. So at increasing concentration of ammonia, we can see that the gas responses increases. We have also developed prototype device using this developed sensor based on like a comparator and microcontrollers. Next slides. So another application, we also tested this type of sensor for the NO2 sensing, and we see that their performance is relatively better, and the sensitivity can go beyond the ppb levels. Next slides. Further, we developed organic thin film transistors for increased sensitivity towards ammonia gases. So in transistor structures, the beauty of this structure is that when we apply a gate voltage, the charge particle increase in the channels. So we can see that increased sensitivity towards gases can be possible in transistor structures. So in transistor structures, we achieved a significant response, more than 50% of gas response. So this is the beauty of transistor structures. Next slide. So further, we increase the gas sensitivity towards ammonia using the composite of CDSC quantum dot with the organic uh, PQT. So when we increasing the composite, that is CDSC quantum dot in the organic framework, the sensitivity towards the gases increases. Next slides. So in next slides, we can see that the sensitivity increased up to 50% in the case of PQT and CDC composite. Next slides. So in this, we achieve gas response up to 90% when you use PBTT polymers. So in this, we develop PBTT polymers using uniform floating film transfer methods, and their interaction increases due to well-oriented PVT film, as well as their better self-assembling in this case. So these are some topic we have discussed today about biosensing applications. as well as gas sensing application. These are crucial for the study of a health monitoring devices or like a biosensing devices nowadays, because these organic based devices will be the future devices, because these are more flexible and based on our used. These are portable. Nowadays, portable devices are very demand, and without development in these organic devices, we cannot imagine the future of flexible devices. So next slide. Finally, I thanks all the collaborator, my mentors, PhD mentors, postdoc mentors for guiding me, and all the alma maters, employee, past employee, and institute I have visited. Thank you very much. I will be happy to answer any query.
Thank you, sir, for this very insightful session. I request all the participants to kindly interact with our speakers. You can ask your queries or discuss your topics of interest to make this FDP a collaborative success. You can now proceed with the Q&A session and ask sir any kind of doubts that you might have. You can unmute yourself and you can ask questions.
Hello, sir. Namaskar, sir. Chandan sir,
Yes,
namaskar.
Yes,
Sir,
yes.
nice and informative session. Sir, I'm having a
Yes.
question that what about the biosensing application, what you have shown? Basically, in your slides, a lot of data you have shown, but can you show, can you give some examples of biosensing items? Mean how one can think these capacitors or these kind of materials, what you have synthesized in your postdoc or PhD for the healthcare applications. Basically, I'm from healthcare department, so I think that if I can listening and I can get some knowledge, so I can think to apply your area in my area. Can you help in that? Bring
Yes,
some
sure,
knowledge.
sure. So in health sector nowadays, we examine bloods. So we can see that the bloods, when there is a change in human body, so significantly we can see that change in urine and blood. Therefore, we most often examine human urine and blood for examine the disease in the human. So these developed biosensors examine that unwanted quantity, like unwanted species in blood or either in urine. So, for example, if there is a lead found in bloods or like urines, or we see the increase in mercury, or maybe increase in like a sodium or potassium. So these increased amount of that ion can easily be detected using these organic devices, and this is a single strip based devices, similar like we nowadays measure sugar. So sugar is also a we measure strip based. So these type of devices now sugar is the most common now people using. So similar type of things in future, people can use blood sodium, blood chlorine, like a sugar detector. That is possible with these type of
No,
devices.
you are right, sir. You are right. But my question is that when you will design the your any sensor, so what you people basically think taken into consideration that whether the zinc has been increased or sodium has been increased or decreased. So you will design that molecule like you have shown one molecule here, the organic molecule that is having some
Yes.
application, right? So
Yes.
what is your thinking in that? Mean that how you are thinking that this can trap the molecule, or it is randomly you are going to put the application on different ions? Is my question. You understand my question? I have.
No, yes, I understand. But
Yeah.
we generally go through the problem. First, I focus on problem. What is the
Yeah.
problem we have to study? Like a, for example, we have to study to detect like mercury in human body, human blood. So our focus is the any material that interact with mercury and their change in electrical quantity. So we focus on that, developing that material that can
If you could
interact with
sir,
mercury. and
Yeah,
give
Chandan
significant
sir.
electrical quantity. Yes.
Chandan sir, if you could share
Yes.
few research papers relevant to this, what is my question to the Gaurav sir? I shall be thankful. Because if
Sure,
Gaurav
sure,
sir
sure.
will share in the group, we can read it. No, please, sir.
Sure, 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 sure. I will
Please,
I
sir.
will share the yeah.
Okay, please, sir.
Thank
Thank you.
So, sir, to add up to Gaurav sir's, actually, to your question, it is like, for example, ZNO has O group on the top, okay? So, the interaction we can see, first we need to see the chemical bonding. The O oxide group can, whether it can bond to some particular compound or not, we need to first analyze. If we are happy that it can bond to a particular group, then we can concentrate on selectivity, interference study, and other things. Based on that only, we will be getting to know whether that particular thin film, whether it will be sensitive to that particular analyte or not. Okay. I hope I
Sir, but
answered your question.
sir, but question is that no, sir, if a normal person is there, in the normal person also lot of minerals are there, right? Zinc, sodium, ions, lot of ions are there. So if the molecule is there, it is having the ability to either coordinately binding or any way it is binding to the ions. So in normal person also ions are there. In deficient or excess person, excess mineral mean hyperkalemia or hypernatremia. In that case, the sodium levels are increased. So there is a affinity of the molecule that it will bind, whether it is a normal level or it is the increased level. So how the
Yes.
molecule is
Yes,
going
yes.
to sense that one? Because in
I
in vitro,
understand, sir.
yeah,
The,
okay, yeah,
I
You are
understand that.
talking about more
Actually, you
towards
are
selectivity, right?
interference. You
yes,
are
yes,
talking
yes,
about
yes, you're right.
interference.
Yeah,
Most of
yeah.
interference. So for this, we generally study selectivity. So
Yeah, yes, sir, yes,
all
sir.
the possible interfering species are also investigating before the reporting. So with
Yeah,
that interference, what is the response? Because materials are insensitive, materials do not differentiate between these are like a chlorine or like a mercury. They only increase ions. So we just investigate all the interference substance that affect the performance. So after a study interference only, we conclude that the sensor are basically useful for that purpose.
yeah. Please, sir,
It is very
please, Uvanesh
important
sir
to
and
study interference.
Chandan sir, both of you, please, I'm requesting you to, if you do have some articles, please share to the Gaurav sir.
Sure,
Gaurav
sure.
sir will share it and later.
Different.
Please, sir,
Okay,
please,
okay,
sir.
okay. Not a
Thank you,
problem.
sir.
Ah.
Any query more?
Is it Sandeep Kumar Tiwari, sir? You can go on. Ask.
Good morning, sir. Good afternoon, sir. Am I audible?
Yes,
Yes.
yes.
Okay, sir. So as I was asking, I'm asking just a simple doubt, sir. Actually, I was working on metal analysis in bioremediation field. So I have a small question. So I have some potent bacteria that are capable of remediating that metal. So what I'm
Yes.
trying to develop is a new biosensor that can detect its presence in the environment, so that we can go ahead with the remediation measures. So what I want to know is, how am I going to integrate that AI and ML part, that specifically that part which help me to use that potent bacteria in this remediation field. I cannot find a beginning step in this process. So if you can help me guide in this.
These are then generally two parts. First part is like a development of devices. First, first you should develop devices, and then data collected from these devices used for the AI ML purpose. So AI ML normally do not work on the hardware; they work on the like a data parts.
Okay.
So, so detection of that protein or like these should be using devices like electronic devices or like chemical based, and this data should be analyzed using AI/ML. Or based on that AI/ML, the best suitable results can further develop.
Okay, so you are suggesting me to first find that collaborative data and then have a
Yes.
proper upper and lower bar so that I can set that limit. And based on that, we can use
Yes.
the software for integration.
Sure.
Okay, sir. Okay, thank you so much, sir.
Any further questions? Any further questions? Okay, I think we do not have any further questions from the participants. Now, I thank Dr. Kumar a lot for agreeing to give the speech in our FDP. And we thank you, sir, for your very insightful and highly informative lecture. Your deep expertise in materials and for biochemical sensing have truly enriched our understanding of the subject. We now take a moment to pay our respects to Dr. Kumar using a virtual memento as a symbol of our gratitude. We request Dr. Kumar to kindly accept the virtual memento from Dr. Gaurav Kumar and highly oblige. Melody.
Thank you, Chandan sir, for accepting the invitation, sir. Kindly accept this e-memento as a token of gratitude from Galgotias University and MNNIT organizing committee member.
Thank you very much, sir. Thank you very much.
Hello.
Thank you very much, sir. I'm
Chandan,
audible,
sir?
sir. Thank you very much, sir, for inviting me to this platform. I'm audible.
Moving forward, we greet our next speaker for the day. It is our pleasure to welcome Dr. Dheeraj Ahuja from MNNIT Allahabad, whose pioneering work in biomedical polymers has significantly contributed to the development of biocompatible and functional materials. Dr. Dheeraj Ahuja holds a PhD and Masters of Engineering in Chemical Engineering from Dr. S.S. Bhatnagar University, Department of Chemical Engineering and Technology from Punjab University, Chandigarh. He completed his BE in Chemical Engineering in 2007. Currently serving as an assistant professor in the Department of Chemical Engineering at MNNIT Allahabad, Dr. Ahuja brings over 8 years of teaching and research experience. His areas of expertise include polymer nanocomposites and bionanocomposites, engineering, bioprocess technology, and biomass utilization, with a strong focus on sustainability and innovation in chemical engineering. He has till date published 13 peer-reviewed international research articles, 6 book chapters, and 1 conference paper. Dr. Dheeraj Ahuja has actively participated in over 20 national and international conferences, presented his research, and has completed 14 workshops, short-term training programs, and faculty development initiatives. His scholarly work has been recognized with multiple accolades, such as research awards for publications in SCI and SCIE index journals, an award of excellence for poster presentation at PSC 2010, first prize in oral and poster presentation at Anveshan Research Convention held in 2010 at the university level. Dr. Ahuja continues to engage in cutting-edge research focused on sustainable materials and advanced chemical engineering applications. Today, Dr. Ahuja will be speaking on polymers for biomedical applications. Sir, we warmly invite you to begin your session. Thank you.
I request all the participants to mute yourself. Because it's like it is giving disturbance. Participants, please mute yourself.
Good afternoon to all. Thank you, Sumedha, for a wonderful introduction. I will be talking on polymeric biomaterials for biomedical applications. Am I audible to all?
Yes, sir.
Okay, thank you. As we all know that in a biomedical era, we are using metal alloys which can be toxic and non-degradable that need to be replaced after the recovery. So there is an urgent need for transformation from alloys to the polymeric material, and that too for the biodegradable polymers, as they can be used as they are biocompatible and non-toxivable. So polymeric materials have various applications in bone tissue engineering, skin tissue engineering, cartilage, cortinal, cardiac, and drug delivery system also. What will be the objectives which we will be accompanying after listening to the after from this lecture? This lecture is focused on the understanding of the fundamental principles of biomaterials. What are the various key properties and characterization techniques, various biomedical applications, and what are the recent advancement and future trends in highly efficient polymeric materials which are being used nowadays? Why we are using polymers in biomedical application? As I already told, the basic need is the biodegradability and biocompatibility. Apart from this, other advantages are also there, like they have versatility in properties, like they have good mechanical properties, along with their good tensile strength. They have good percent elongation, so that they can be stressed to the extent they are required. In heart valves, we use silicon balloon, which can expand because of its percent elongation. Then its chemical properties, its physical properties, they are light in weight. One more important advantage of using polymers is that they have the tunability in properties, like we can vary their molecular weight, cross-linking, and functionalization by varying the concentration. As we can take, if we take the example of polyurethanes, in that we have two segments, soft segment and hard segment. By varying the concentration of the soft segment and hard segment, we can tune the mechanical properties and its cross-linking. Further, polymers can be easily processed and fabricated. They are easy to manufacture products with different shapes. First, we will start with the basic. What is a polymer? As we all know, it is the repetition of the small or repeating units, which is called monomer. And monomer is a small molecules or building block. Like, for example, if we take the example of PVC pipes that we use in our day-to-day life, that are made up of vinyl chloride, in which one monomer repeating unit is vinyl chloride, which is repeated for polymerization process, and it is called vinyl chloride. Then we will deal with degree of polymerization. What is degree of polymerization? Actually, number of repeating units. As we know that we repeat the monomer, various monomer units are joined to form a polymer, and the joining unit, monomer unit, is known as repeating units. As the number of monomer units we have in a polymer is known as the degree of polymerization. And from the degree of polymerization, we can calculate the molecular weight of a polymer. How? Suppose we have ethyl, for example, we have vinyl chloride, and if its molecular weight is like 60, then we have used 10 degree of polymerization, 10 units of vinyl chloride are used, then its molecular weight will be 6000. In the same way, we can calculate the molecular weight for any of the polymers using degree of polymerization and knowing the molecular weight of the repeating units. The molecular weight generally varies from 10,000 to 1 lakh Dalton atomic. For making the polymers, we use different polymerization process, generally condensation and addition polymerization, which are condensation polymerization reaction is also known as step reaction polymerization, while addition polymerization is known as chain reaction polymerization. In addition polymerization, we linearly add monomers one by one, while in condensation polymerization, monomers are joined by eliminating the small molecules like water vapor, water molecules, hydrogen chloride, and then ring opening polymerization, which is most widely used in the current scenario for the formation of diols, for the formation of ring opening like polyesters. In this, cyclic monomers are generally linked to a linear polymer chain, and in this, cyclic compounds are formed. For example, like polylactide, I told polycaprolactone diols are formed nowadays for forming polyurethanes, and polyurethane is used for like stunts, sutures, other biomedical application. It is most widely being used. Then as we proceed, we have the classification of the polymers. Polymers, on the basis of their chemical structure, physical properties, mechanical behavior, thermal characteristics, can be classified as natural and synthetic polymers, organic and inorganic polymers, thermoplastic and thermostatic polymers, and apart from this, plastics, elastomers, fibers, and liquid resins. Initially, we will study basics about these polymerization techniques, like what are natural polymers. Natural polymers are most widely used in the biomedical arena, and they are derived from living creatures and are of great interest in biomaterial field. And the advantage of using natural polymers is because of their natural properties, that is, they are biodegradable, non-toxic, mechanically similar to the tissue which is to be replaced from the human body, and they are highly porous. Some of the examples of natural polymers that are being used in the current research are collagen. Its major component is it is the major component of connective tissue, and it is used in scaffolds and wound dressings. Cellulose, it is derived from plants or waste biomass like agricultural residue, and it is used for wound dressing, drug delivery application. Then alginates, then alginates, they are obtained from brown algae, and they are used in hydrogels, cell encapsulations. Gelatin, it is denatured collagen, and it is used in drug delivery, tissue engineering. Then from chitosan, it is derived from chitin, antimicrobial, and it is used for antimicrobial application, hemostatic, and drug delivery applications. Then we have synthetic polymers. Synthetic polymers, because of their good mechanical properties and other advantage, is most widely used. But we are replacing it because because of their degradability issue, as organs need to be replaced again and again. So these are low molecular weight compounds, and their examples are polythene, PVC, nylon, polypropylene, polycaprolactam, polymethyl methacryl, and terylene. They are reproducible and have mechanical properties as compared to natural polymers, but they have the issue of biodegradability and biocompatibility. Some of the advantage of using synthetic polymer is their ease of manufacturability, processability. They are available at reasonable cost, and the required properties like physical properties, sterilizability, and manufacturability. They are widely used at present. They are being widely used in various applications, like for preparing medical disposable items, prosthetic materials, dental materials, like implants, dressings, polymer drug delivery, tissue engineering products. The polymers are classified into four. Synthetic polymers are further classified into four categories. One is biostable polymers, bioerodible polymers, water polymers, and other polymers. Biostable polymers, as the term biostability suggests, it is the stability of biomedical polymers in physiological environment as a function of time. Like if we are replacing using biostable polymers, for how much time it can be used in our body with stability, and it will not be affected from the other toxic materials, and they are and its application, it is sufficiently used in artificial organs, blood pumps, blood vessel prostheses, heart valves, skeletal joints, and kidney prostheses. Mostly, biostable polymers are used for long-term applications, like for pumping of the bloods as a stunts. So we use biostable polymers in that area. Bioerodible polymers, bioerodible materials that will serve as a short-term purpose in body and then decompose to small molecules that can be metabolized or excreted sometimes with the concurrent release of drug molecules. These types of polymers are used for tissue engineering applications, like when some of the tissue is damaged, or like bone tissue, mainly in bone tissue engineering, when like some bone is broken. Nowadays we are using rods, alloy rods, which are being replaced by polymer rods, which will degrade after some time with and it will be excreted from the excretable material. They are also used. They are also used in surgical sutures and tissue growth materials and control release of drug also. Then water soluble polymers. Usually bioerodible polymers are known as water soluble polymers, and they include form of plasma or hot blood substitute, or which may function as macromolecular drugs. It improves the behavior of pharmaceuticals and used in blood substitute as viscosity enhancers or as oxygen transport molecules. Then other polymers apart from these categories are plastic, elastomers, fibers, and liquid resins. Depending upon the its ultimate form and use, polymer is classified as plastic, elastomer, fiber, or liquid resin. If a polymer is shaped into hard or tough utility articles with application of pressure and heat, then it is termed as plastic, and its common examples are polystyrene, PVC, polymethyl methacrylate. When we vulcanized it into a rubbery product and it has good strength and elongation, then it is termed as elastomers. Typical examples are natural rubber, synthetic rubber, silicon rubber, which are basically used in the heart valve applications. If drawn into long filament materials whose length is at least 100 times its diameter, then the polymers are said to be fibers. And basic examples are nylon, terylene, cellulose, silk fibers. And cellulose and silk fibers are nowadays most widely being used in biomedical applications for making, like when we have COVID-19, cellulose fibers were used for the manufacturing of masks, which have antibacterial properties by the addition of some antimicrobial bioactive compounds into cellulose. Polymers, when used as adhesives, coating compounds, sealants in liquid form, they are described as liquid resins. The commercially available epoxy adhesives and polysulfide sealants are typical examples. One more example, it's polyurethanes, which I am focusing from the starting, can also be used as a resins, and it can be used for the coatings of the it can be used as antibacterial coating agent for wound healing applications, or for your electronics devices or your car interior, like if from where we have, well, mostly contaminated disease we carry from these devices, and it is being used by all the day, so they carry a lot of microbes. So antibacterial coatings can be used in form of resins, and then they can be coated on any of the devices, electronic devices, and car interiors. Then we have organic and inorganic polymers. As its name suggests, we all know that organic polymers and inorganic chemistry and inorganic chemistry. Organic chemistry deals with chemical carbon atoms, hydrogen connected with hydrogen, oxygen, nitrogen. Similarly, organic polymers are those in which the main backbone is carbon atom attached with hydrogen, oxygen, nitrogen. The majority of, and then we have most of the synthetic polymers we are using in day-to-day activities, from day-to-day activities to aerospace applications, are generally synthetic polymers, which are organic polymer, and they are organic. In fact, number of organic and inorganic polymers in which their backbone, carbon backbone, they do not have carbon backbone, and their examples are glass and silicon rubbers. Then thermoplastic and thermostatic polymers, this we all know. As the name suggests, plastic, it is, I already defined plastic, that which we can give it a particular shape with the application of pressure and temperature. Similarly, in thermoplastic polymers, we can convert it into any shape with application of heat, and on cooling it takes a permanent shape, and if we again heat it, and we can change its shape by heating, then it is called as thermoplastic. If we cannot change the shape of a polymers, or once it achieves a shape, then it is termed as thermostatic polymers. But there are some of the parameters which we need to be considered. We need to consider for biomedical, for the application of polymers in biomedical applications. The design and selection of biomaterials depends on different properties. These are host response, biocompatibility, biofunctionality, functional tissue structure, toxicology, appropriate design and manufacturability, mechanical properties of biomedical polymers. Host response. What is the host response? It is the body's local and systematic reaction to implanted polymeric materials or devices, including inflammation, immune activity, healing, or rejection, crucial for implant success. Like in this, we want to say if we are adding some polymers into the body as a bone tissue engineering, as a tissue implant, how it will respond to the body. Like sometimes you would have seen in your day-to-day life, like many of the people around us, which are of old age, went under heart surgery, and they have been put with the stents. Sometimes you would have seen that it is not compatible with the heart, and we feel difficulty after going the surgery, and the doctor gives some medicines and asks that your body is taking time. That is the host response. Then it is biocompatibility. Biocompatibility is a material's ability to function in body without causing harmful local or systematic effects, supporting healing without toxicity or immune issues. Toxicology, it ensures materials and their breakdown products are non-toxic and safe for cells and tissues. Then appropriate design and manufacturability. Before using the biomaterials, we should ensure that it can be easily shaped or machined or molded into the shape we require. So that is the appropriate design and manufacturability we require. Then mechanical properties of biomedical polymers. The key biomedical properties are tensile strength, yield strength, elastic modulus, surface finish, creep, and hardness, and determining polymer performance under physiological stress. What does it mean, determining polymer under physiological stress? Like once with the combination effect of temperature and pressure, like stress, load, and it is used for determining its shape memory applications, which are most widely used in the polymers. Nowadays, shape memory polymers are used for their application as they can get into a shape and then get back to the when stress is applied, they change their shape, and when the stress is removed, they get back to their original shape in which we have. And the most common example of this shape memory polymers is our orthodontic mattresses, which we use at our home for sleeping. When we go for buying a mattresses, we very commonly listen from the marketing person that, sir, it is a orthopedic mattresses, and it will keep your backbone in a shape. Why it is so? Because it is made up of shape memory polymer, and the polymer used is polyurethane. Okay, it is in the form of elastomers, that is foam. And when we sit on it takes the shape, and when we sit on it or when we sleep on this, we apply stress on this, so it takes the shape of the body, and when we get up, it gets back to its original shape as a flat surface. Medical and What are the medical and biotechnological uses of polymeric biomaterials? They are used as cardiovascular system implants and devices. They are used for the repair and replacement of bones, joints, and dental structures, for vision correction through contact and intraocular lenses, artificial kidneys and materials for hemodialysis, membrane design for oxygen transport, surgical stitching materials, polymers that promote tissue integration, system for the controlled delivery of the pharmaceuticals. Some of the key properties of polymeric materials that are required for biomedical applications are biocompatibility, biodegradability or bioresorbability, mechanical properties, chemical properties. Some of the other important properties include sterilizability, manufacturability, surface porosity, and biomimicry. So we will discuss in detail about these properties like biocompatibility. What is biocompatibility? Is the ability of a material to perform its intended function without eliciting undesirable local or systematic effect in recipient or beneficiary. It is crucial for avoiding inflammation, rejection, and toxic reaction in the body. Some of the common examples of the polymers which are biocompatible are polyethylene. It is used in orthopedic implants like hip joints and in catheters, and it is known for its inertness and long-term stability within the body. Then polypropylene, it is commonly used for sutures and hernia meshes, also valued for its biocompatibility and versatility. Then silicone, it is used in the form of a rubber and find use in finger joint prostheses, maxillofacial implants, and artificial bladders, gallbladders. Because of its flexibility, chemical inertness, and biocompatibility, it finds so many of uses. And another example is polytetrafluoroethylene, which is commonly known as Teflon, and it is used in cardiovascular graft, sutures, and epidural catheters because of its chemical stability and low friction properties. Then polymethylacrylate (PMMA), which is rigid, transparent, and it used in bone cements, intraocular lenses, and dental applications. Apart from this, if we PMMA is also used in the manufacturing of diapers, which are used for the kids. Then polyethylene glycol, it is value for it is because of its hydrophilicity and lack of immunogenicity, it is used in drug delivery and surface modification of various polymers for their wide applications in different area of biomedical. Yeah, then other properties, biodegradability or bioresorbability. It is the ability of a material to degrade within the body after performing its intended function and be absorbed by the body without leaving harmful residues. It is essential for temporary implants like sutures, drug delivery, and tissue engineering scaffold that are designed to disappear as the tissue regenerates. The most common example of biodegradable scaffolds is polyurethanes made from bio bio-based polyols like vegetable oils, lignin polyol-based oils, polyols, cellulose-based polyols, and uh, without diisocyanate materials. And some of the common examples for biodegradable polymers are polylactic acid. It is biodegradable and bioresourceful polyester, extensively researched for application in drug delivery, sutures, and tissue engineering scaffolds like orthopedic implants. Then polyglycolic acid, it is biodegradable polyester and is used for sutures due to its high crystallinity and impressive strength. Both PLA and polyglycolic acid, they are used for drug delivery or suture application because of their hydrophilicity. And another example is they have different molecular weights, like we can take it from low molecular weight to high molecular weight, and because of its like variation in its molecular weight, we can tune the properties of the resulting polymers by using these polymers. So accordingly, we can adjust properties. So these are two most important biodegradable polymers, which are most widely used nowadays in the recent arena of research. Then polylactic co-glycolic acid, it is a copolymer of PLA and PGA and offers controlled degradation rates by adjusting the ratio of lactic acid to glycolic acid, and it is used in drug delivery system and tissue engineering. And then polyecaprolactone, I already told it is a synthetic polymer, but it is not hydrophilic, it is a hydrophobic, and it has semi-crystalline with a long degradation time. It takes a long time to degrade as it is a synthetic, as it is a as it is a synthetic polymers. So we used it for long term implants basically. Then chitosan, it is a natural polysaccharide known for its biocompatibility and biodegradability, as it degrades easily. So it is used for wound healing properties or in drug delivery systems. Then collagen, it is also a natural protein, and it is also used for tissue engineering scaffolds, and it helps in promoting cell adhesion and proliferation. And then another properties is mechanical properties. It includes tensile strength, compressive strength, flexibility, stiffness, hardness, and toughness, which dictate a polymer's ability to withstand forces and deformation. We will be dealing about the detail in the coming slides. Need to be matched for specific application. For example, we take like joint replacement. We require high strength and fatigue resistance, while flexible materials like silicone are preferred for catheters or contact lenses. Its properties can be tailored by adjusting various factors like molecular weight, cross-linking density, crystallinity, and copolymer ratio, as I already told in the previous slide about PLA and PGA, which can be used, which are most widely used because by adjusting, they have different molecular weights, and by adjusting different molecular weights, we can tailor their properties. Then polyether ether ketone, it is a high performance polymer used in load bearing orthopedic devices like knee joints, spinal fusion, owing to its mechanical properties closely matching to those of natural bones. Then polyurethanes. Polyurethanes are elastomeric materials, I already told, and they have versatile application in the area of biomedical, not only in as it has application in all of the areas of the, from like taking day-to-day activity of home to the aerospace application. Like in home, we use it in the form of forms, in the form of shoes. The shoe sole is made up of polyurethanes, like the seat covers which we sit, the foam inside it is made up of polyurethanes. The paints, the plastic paints which we hear nowadays, or PU paints we hear, that is polyurethanes. So it has a wide application in the all the world. Similarly, it has a wide application in like biomedical area also. It can be used as a stunt also. It can be used for tissue replacement also in tissue engineering. It can be used as a hydrogel for drug delivery application. So it has a wide application. Then ultra high molecular weight polyethylene offers excellent mechanical strength and wear resistance, making it suitable for long-term implantation in joint replacement. Whenever we have to use joint replacement, we try like a hip replacement, or basically in hip replacement we use UHMPV wires, which we will be discussing in the coming slides. And then chemical properties includes like chemical composition, structure, like whether it is a linear polymer, it is a branched polymer, or branched or network polymer. Then its crystallinity, then chemical resistance, its glass transition temperature, and interaction with the surrounding environment. The most important role played by is glass transition temperature and their structure and chemical composition, and for varying its properties. Then the degradation behavior, stability in body's environment. It can be modified by altering monomer composition, introducing functional group, and using surface modification technique. Then hydrogels, they are made up of like polyvinyl alcohol, PVA, agar, alginate, and they absorb large quantities of water and mimic the structure of soft tissues. And they are used for wound dressing, cell delivery or drug delivery, tissue engineering scaffolds. Then alginate, it is I already know that it is a natural polysaccharide. It is also used in the form of gels in the presence of divalent cations, and making it useful for wound dressing, drug delivery, and tissue engineering scaffolds. Some of the other important properties that we should take care while deciding the polymer for biomedical application is sterilizability. Polymer, while selecting the polymers, we should take care that as sterilization is most important parameters to be considered for biomedical application, as all the things we use is sterilized before using it again. So we should take care that it should be sterilizable, that is, it is autoclavable, resistant to radiations, and it without degrading or becoming toxic. Then its manufacturability, that it should be easy. It can be easily manufactured into different shape, and and then surface properties like its surface energy, roughness, hydrophilicity, hydrophobicity, influence, cell adhesion, protein absorption, and interaction with the surrounding tissue or fluid. So it is an important property we should take consider. Some polymers can be designed to mimic the properties of the body's natural tissues like extracellular matrix. So these are some of the other important properties we should take care. Then the characterization techniques which are used for the characterization of polymers are like for determining the molecular weight, we use gel permeation chromatography, size exclusion chromatography, Maldi-Toff MS spectroscopy techniques. These are some of the techniques which we use for determining the molecular weight of the polymer. Why we determine the molecular weight? As I already told, it plays a vital role in the in designing the in deciding the mechanical properties of the polymers. So it is very necessary. Sometimes after knowing the molecular weight, we can proceed further for by making the molar balance for making the for the manufacturing or for the synthesis or designing of the polymers. So it is an important parameters to be decided. So gel permeation chromatography, size exclusion chromatography, Maldi-Toff MS spectroscopy is most widely used for determination of molecular weight and molecular weight. Then nuclear NMR and FTIR, it is used for the confirmation of chemical structure. Like if our we are making the like we are making our polymers, whether it is whether uh, it has taken whether our reaction of the polymer, like for making a polymer, we do polymerization reaction in which two or more polymers are added to verify whether the reaction is whether the reaction is completed, and the actual all the completion of reaction and the formation of the structure. We use NMR. NMR is of three types: HNMR, proton NMR, which is known as proton NMR, then carbon NMR, and then phosphorus NMR. These three type of techniques are used for determining the chemical structure. Then FTIR, we will discuss about NMR and FTIR in detail in the coming slides. Then thermal characterization. Thermal characterization is done in two ways. One is thermogravimetric analysis, that is for determining thermal stability and degradation profile, and another one is differential scanning calorimetry. It is generally used for determining the glass transition temperature, melting temperature, and crystallinity. Then mechanical properties, it is determined using universal testing machine Instron, and we get from this we get stress strain curve and compression curve, and we get the values of tensile strength, compression strength, flexural strength, Young modulus, elasticity. Then dynamic mechanical analysis, it is done for determining the viscoelastic properties, like how it behaves under stress and temperature, and whether viscoelastic means it has elasticity along with viscosity. Highly viscous material which has some properties of elastic nature, that is known as viscoelastic polymers. Then fatigue testing means resistant to cyclic loading. Cyclic loading means like initially if we are taken load from 1 to 10 and then 10 to 1 with the application of along with variation in its temperature, like we are varying the temperature from room temperature to 250 degree, then 250 degree to room temperature. It is the cyclic condition. Like in cyclic loading, what happens to the our mechanical properties, that is known as fatigue testing. Then we do surface and morphological characterization. It is done using surface. It is done for determining surface morphology and porosity using SCM. Then atomic force microscopy. It is used for determining the topography of the surface at nanoscale. Then X-ray photoelectron spectroscopy (XPS), most widely used technique nowadays for determining surface and elemental composition. Then contact angle measurement, it is used for determining surface wettability. From here we get a contact angle from which we can determine that whether our compound is a hydrophilic or hydrophobic. And then biological assessment, it is in vitro and in vivo. In vitro we do cell viability, proliferation, differentiation, gene expression, and in vivo we do animal models for biocompatibility, tissue integration, and functional outcomes. some of the analysis and testing of polymers, like chemical method of analysis. It is two powerful techniques for analyzing low molecular weight products of reaction of polymers are mass spectroscopy and gas chromatography. Mass spectroscopy is used for low molecular weight fractions, which are non-volatile compounds. And for volatile compounds, we use gas chromatography. Like for gases, we use gas chromatography, and for other solids and liquids, we use mass spectroscopy. Then we use infrared spectroscopy. In infrared spectroscopy, which we studied about FTIR, Fourier transfer infrared spectroscopy. In this, we have stretching vibration, bending vibration, and the combination of both the vibrations. And they may be symmetric or asymmetric, scissoring, in plane bending, rocking, wagging, or twisting. And we get in this, we test generally we test from in the region of 4,000 to 400 centimeter inwards. And on the basis of the peaks, we get the class of compounds, and it is the table of class of compound is shown in the slide. And then from FTIR, we get generally this type of curves, and these are the numbers like 2922, 2849. These are the peaks from which we can, like here you can check from this peak. The peak between 2850 to 3000 is due to alkanes. From these peaks we can calculate all the things. We can determine which bond is present, our reaction is completed or no, not completed. These are the regions shown are for these curves. FTR spectrum shown in these figures are of lignin and soda lignin, and ETR and ETA, soda lignin and modified soda lignin. Then nuclear magnetic resonance. It has become a powerful technique for chemical and magnetic analysis of samples. It is used for sequence distribution and micro structures in polymers. It is generally based on it generally works on the principle of MRI that we use for use for our system head systems for determining like we have we do MRI of various diseases. So in the same principle, it depends and it determines the molecular. It is generally used for the identification of polymers or functional groups. It monitors progress of polymerization process, whether the polymerization process is completed or still some time is required, its composition, chain molecular structure, type of polymers, toxicity, and end groups. I already told it is of three nuclides, which it contains hydrogen, carbon, nitrogen, oxygen, phosphorus, fluorine. Chemical shift of some prominent groups of polymers, like this peak is shown for lignin and modified lignin, which we are known as polyol, which has been used for the development of polyurethanes for biomedical application, as lignopolyol is degradable. Degradable, because it is made from natural polymers, that is biopolymer, not polymer, natural polymer. It is made from biopolymer. Lignin, second most abundant biopolymer on the earth. And this we have shown that range, if our peaks are within the range of 0 to 1 ppm, then they are alkanes. 4.6 to 5.8 ppm, it are vinyl groups, and 2 to 3.5 acetylenic group, aromatic groups, alcohols group, phenol groups, depending upon their ppms. In the figure shown, you can clearly show for EL, the spectrum shown of EL, we have peak at in the region of 2 to 3, 4 to 5, and 6 to 8. However, in the region of PO, we can see that the peaks which are confronting to the aromatic, like 6 to 8, we have 3 peaks. One is between 6 to 7, one is for around 7, and then one is around 7 to 8. And these 3 peaks, where we have this structure on the structure of lignin. Lignin basically consists of 3 aromatic units. And when we modified it to lignopolyol, its aromatic structure break down into linear structure. And it is like it is shown in through the NMR structure. Then, as we were discussing about mechanical properties, this week you can see in the graph, like it is the maximum strength where a polymer break. It is known as the strength, and how much it is elongated during the length. It is known as the elongation at a break. and stretching. There are several types of strength, like tensile strength, how much it can be strength. Compressional strength is basically used for the forming materials, bottles. Then like how much we can compress that polymers. Flexural strength, how much we can bend some of the polymers. Torsional, twisting of the polymers. Impact means hammering and so on. And we know that the strength, well, we have different structures of the polymers, like linear polymers, branch polymer, cross-linked polymers, and network polymers. And their strength structure, for strength for network structure is the highest. That is why if we take example of cellulose and lignin as a filler, they are being used nowadays as a natural biopolymer, or in their nano dimensions, for the development of the nanocomposites for biomedical application. Because of their network structure, they have good mechanical properties. How the mechanical testing is done, there are several standards for testing different types of plastic. Like for testing of plastic, we have the ASTM the standard D638. Then for galvanized rubber, we have D412, and for thin plastic sheeting, we have D882. And for one thing we have to take care that for, like for testing of the tensile strength, or we take tensile specimen in dumbbell shape, as shown in the figure, with width and thickness known, and we put the width and thickness, and it is the assembly in which, like this type of samples, it is shown, and the result obtained is like stress strain curve we obtain. Like it is shown where its failure is happening, it is at the tensile strength, and then ultimate tensile strength, elongation, modulus, toughness. These properties are calculated. How these are calculated will be shown. Then the characteristic feature, like if peak is soft and weak, then the modulus is low, yield stress is low, ultimate strength is low, elongation at break will be moderate. If the polymer is soft and tough, how we will determine that it is soft and tough? If its modulus is low, if yield stress is low, ultimate strength and elongation break, both are strength and break elongation are high, then it is soft and tough. If hard and brittle, then its modulus will be high and ultimate strength will be moderate and elongation will be low. Then hard and tough, it's all the properties, modulus, yield strength, ultimate strength, elongation strength, all will be high. And we can also see, like from the graph we obtain, we can come to know like what type of material it is. Then DSC, it is generally used, I already told, it is used for determining glass transition temperature, melting temperature, crystallization, crystallinity, and it generally measures the heat flow under isothermal conditions. And it can be used for the characterization of polymers, fibers, elastomers, composite film, pharmaceutical materials like drugs, foods, cosmetics. It is the apparatus. In this, it is connected to a computer in which sample pan is in a pan. We keep our sample and seal it and put it into the reference, put into the heater. With simultaneously a reference pan is heat, and we test it into two sealed pans are simultaneously observed for determining the changes, like it's a reference pan and a polymer pan. Then we set the parameters. Then three parameters, like glass transition temperature. What is glass? It is the temperature below which polymer is in the glassy state and above which it attains rubbery state. So it is most widely used for determining the state of the polymer, where the material will turn into different shape. As if we all know that late back, when Titanic was formed, it sink into the sea, because that was the main reason for that, was that as accidentally it went into the cold environment, cold water, the temperature of the water was very at that time we came to know about the concept of glass transition temperature, because at that time when it accidentally went to the cold temperature, the metal break, and the water come inside the ship, and it sank. So it was the main concept about the glass transition temperature from where it started. It is the typical DSC curve that we generally obtain. And there are three peaks. One is for glass transition temperature, one is for crystallization temperature, and one is for melting point. It is temperature, and it is plotted between heat flow and temperature. These are some of the properties which we can calculate, like heat capacity, glass transition temperature, crystallization, then melting point. And this is all we have put together. Then polymer crystallinity. Polymer crystallinity can be measured by the area under the curve of melting curve. For this, we plot a heat flow versus temperature diagram, and we calculate the heat into temperature upon time into mass. From here, we can calculate the crystallinity using DSC curve, and degree of crystallinity in percentage is given by multiplication by 100, where heat of diffusion determined from DSC thermograms and heat of fusion of 100 plus crystalline sample. These are some of the case studies which I have shown. Then it is thermogravitic analysis, TGA, and from this we can determine highly volatile matter content, medium volatile matter, combustible material, ash, and mass loss. Thermogravity provides information regarding polymerization reaction, efficiency of stabilizers and activators, the thermal stability of final materials, and direct analysis. Provide a journal technique to determine the amount of highly volatile matter, medium volatile matter, combustible material, and ash content of compounds. This method is useful in performing a compositional analysis in polymers, and it can be applied to solids as well as liquids, but cannot be applied for gases. It is the apparatus. In this, we keep the sample in the pan. After it weighs automatically, and with the increase in temperature, as the heat increases, its mass starts to decrease. Its mass start decreasing. Then here, how we can calculate the high molecular weight content, low molecular weight content, medium volatile matter content. From the same the graph obtained, we can plot the DTGA curve, that is differential thermogravitic analysis, first order and second order. This is the case study how tire elastomers, it is went like initially it is showing for oil, then for polymer, and then for carbon black. Three degradation peaks are shown, like elastomer contains oils, polymer, and carbon black. Like in elastomer, three things are present. The peak for elastomers is seen around 200 degrees Celsius. Then for polymer, it is seen around 380 degrees Celsius. And for carbon black, that is ash, when the material is almost fully burnt, it is seen around 700 degrees Celsius. This is thermomechanical analysis. It is used for viscoelastic behavior for the. This is the case study. Then applications of polymers in biomaterials, biomedical implants and devices. We can use polymeric materials in orthopedic implants like polyethylene, polypropylene, polymethyl methacrylate. These I already told, these all are used for artificial joint bone fixation, and the wire ultra high molecular weight polyethylene. We have used this in this way. We can, like plastic liner, which is, it is made up of ultra high molecular weight polyethylene, and it is used as a hip replacement polymer. Then cardiovascular devices, we have polymer material like tetrafluoroethylene, polyethylene, tetramethylate, and polyurethane are employed in. These are used in heart valves, vascular grafts, and pacemakers, implantable defibrillator, catheters, and tubing. Then ophthalmic devices like contact lenses and intramolecular lenses. Then dental application find use in dentures and dental implants. Fillings, like most of the fillings are used made up of zinc oxide. Dentures are basically made of PU. Then soft tissue repair and replacement, like sutures, biodegradable polymers like polyglycolic acid, PGA, PLA, polylactic acid are widely used for surgical sutures, hernia meshes, breast implants. Drug delivery system control and drug delivery system control systems are used for this. Polymer matrices are encapsulated into drugs and then released at a controlled rate over a prolonged period, improving drug efficacy and reducing side effects. Its example include microparticles, nanoparticles, and films. Drug delivery can be achieved in three ways, like targeted drug delivery, implantable drug delivery system, polymeric drugs, and prodrugs. Targeted drug delivery systems, polymers can be functionalized with targeting moieties to selectively deliver drugs to specific cells or tissue, particularly in cancer therapy. If we have to deliver a drug at a faster rate to a particular specific location for treating of that particular disease, we take a polymer in nano dimensions as its surface to length ratio is high, as that is its aspect ratio is high. So it can be targeted to a particular site with a maximum recovery in a shorter time. Then implantable drug delivery system. Biodegradable polymers are used to create implants that release drug over an extended period, eliminating the need for frequent administration. Commercial example include Vitrasert and Ozurdex. Then polymeric drugs and prodrugs. Some polymers are inherently biologically active and can be conjugated to drugs to form prodrugs that release the drug at target site. Then tissue engineering and regenerative medicines. In tissue engineering, they are generally used as scaffold for tissue regeneration. And then wound healing and dressings, cell and gene delivery. In scaffolds, porous polymeric scaffolds provide a 3D environment for cell growth, proliferation, and differentiation. They can be fabricated from natural polymers like collagen, gelatin, chitosan, silk, alginate, or synthetic polymers like PLA, PGA, polycaprolactone, and their application include regeneration of bone, skin, nerves, and blood vessels. Then wound healing and dressings. Polymeric materials nowadays are most widely being used in wound dressings because they absorb exudate, protect against infection, maintain a moist environment, and promote tissue regeneration. Most widely used, I told that they are polymers are generally electrospan nanofibers are most widely used nowadays, made up of cellulose for wound healing applications. They include cellulose, chitosan. They are most widely used electrospan nanofibers or cellulose acetate. They are most widely being used in wound healing application nowadays. Then cell and gene delivery polymers used are utilized as carriers for delivering cells and genes to specific location in body for therapeutic purposes. Nanoparticle-based systems are particularly promising for gene delivery, offering advantages like robustness and efficient cellular uptake. Other applications include biosensor and diagnostic devices. They can be incorporated into biosensors and they can to detect disease biomarkers, monitor physiological parameters, and facilitate medical images. They are also used most widely. They are being used in for the medical devices packaging in form of polyethylene, polypropylene, and polyethylene tetrabethalate, offering protection from contamination and damage. Polymeric biomaterials. This is the research work which we have done in our laboratory. So polymeric biomaterials for bone tissue engineering. Like, what is the main objective of tissue engineering? Tissue engineering aims to regenerate damaged tissue instead of replacing them with grafts by developing biological substitute that restores, maintains, or improves tissue functioning. And other goal is to alternatively act as natural ECM for the growth of new functional tissues in vitro and in vivo. It is some of the example why tissue engineering is used again and again nowadays, and most widely we are exploring new technologies for tissue engineering because millions of people are suffering for tissue or organ loss due to some diseases or accidents, mostly due to accidents every year in around the globe. Yearly cost for this tissue replacement is around 400 billions. Only major medical treatments available in organ transplantation, shortage of replacement tissues, need for alternative sources, development for transplantation by engineering tissue. Some of the characteristic biomaterials used in tissue engineering This I already told these techniques which we should take care. One of the techniques we are using is ceramics. Ceramics are most widely used as a drug delivery in organic non-metallic materials that can either be bioinert or bioactive. Various ceramic materials used in this category are alumina, zirconia, hydroxyapatite, bioglass, glass ceramics. Among all these materials, hydroxyapatite, because of its similar structure, like calcium or mixture of calcium and phosphate, which have good application in bonds. It is most widely used, and it is made up of tricalcium phosphate. Hydroxyapatite is a complex phosphate of calcium, so it has advantage of thermodynamically more stable. Its molar ratio of calcium and phosphorus is around 1.67. That is very good, and it has good osteoinductive and biocompatibility. It can be easily reinforced into any of the polymers for enhancing mechanical properties along with the addition of its bioactive properties. Natural sources of hydroxyapatite are bovine bone, eggshell, limestone, fish waste, orange peel, red algae. Some why polymeric biomaterials. This I already told you. Why degradable polymers is used in tissue engineering, as they can be used as implant and does not require a second surgical event, as they can be excreted with the drugs, relatively easy to tailor mechanical properties, degradation times, and other properties of synthetic polymers. So we have used degradable polymers in this study. We have used we have synthesized um, synthesis nanohydroxyapatite polyurethane nanocomposite scaffolds using vegetable oils, that is castor oil. We have used initially we synthesize and surface modified nanohydroxyapatite. Synthesis of hydroxyapatite by using calcium and nitrogen, and in the present by maintaining pH using ammonia solution, using sol gel techniques at a pH of 9. After that completion of the reaction, the mixture was aged for 24 hours at room temperature. Then the phase replacement was done, and it was dried at 80 degrees Celsius for 12 hours. Then it was subjected to thermal treatments or sintering at various temperatures, and we get the hydroxyapatite powder. And we have shown the white color. It is the hydroxyapatite powder, and it is the structure of the hydroxyapatite. As from the structure, you can clearly see that it has calcium and phosphate groups, and its SCM is shown in the figure. And then after obtaining the hydroxyapatite, it can we directly use it for the for as a reinforcing agent in the polyurethane matrix. But we found that it didn't show the result as they were expected. So we modified it, grafted it with etitronic acid and triethanolamine. We added etitronic acid, which has two phosphonate groups, and so it gives good results for the enhancement in the strength for biomedical applications. And then we used it for different applications. Synthesis of polyurethane nanocomposite scaffolds. The castor oil was used as a as a soft segment for the polyurethanes. In this hydroxyapatite powder in pristine and modified form was reinforced was reinforced into was dispersed into it using high shear homogenization coupled with ultrasonication, and then it was reacted with BDO and MDI, which is one for BDO is one for butanediol, which is used as a chain extender, and MDI is a diphenyl methane isocyanate used as a hard segment for the formation of the polyurethanes. And stannous octet was used as catalyst, triethanolamine was used as co-catalyst, and water and CO2 as forming agent. And these are the results we obtained. The conclusion of the above project above work was that then hydroxyapatite was synthesized successfully with a dimensions with a size of 10 to 100 nanometers. The best result for TEM and SD nanoparticle observed for sample sintered at, and they were sintered at different temperatures, and 400 degrees Celsius indicate good result. bioactivity and biodegradability of scaffold was 80% with 30% filler concentration and improved compressive strength. Overall, it was shown that nanogenic scaffold showed surface with improved cell growth as observed from in vitro cell culture study, indicating that it can be effectively utilized for rapid in vivo tissue-borne regeneration. Some of the great challenges in polymeric biomaterials are that its long-term integration without chronic inflammation, scaling up the production to complex or high-performance materials, overcoming vascularization hurdles in large tissue reconstructs, reducing cost for widespread accessibility. But apart from this, we have an exciting future ahead. Like if we continue giving new innovations in material synthesis and fabrication, we can be a leader in the biomedical industry. And then deeper understanding of cell material interaction is needed, which we shall do. A shift towards smart and adaptive implants, synergy with genomics, proteomics, and AI, which is most widely used nowadays. And then how we faculties all over can contribute in the field of polymeric materials for the advancement in biomedical arena, like in research, and we can by doing leading cutting edge research project, and which can be transformed into a pilot plant scale or some startup, and then curriculum development, integrating some of the new courses related to biomedical application in polymers, nurturing the students to become the scientist, or giving some some mentorship lectures or motivating lectures to them for inspiring them to go into the research area, or becoming a scientist. Interdisciplinary collaboration, like we should collaborate with the industries, and we should foster and resolve the problems of the industries which are being faced by biomedical industry, and we should work in the with that area. We should collaborate with clinics for the testings and other things with industries. Overall, it is concluded that polymeric biomaterials are at the forefront of medical innovation. Their journey from fundamental science to advanced application continues to transform healthcare. Our role as a faculty is pivotal in driving this progress through research, education, and collaboration. Let's collectively shape the future of medicine with highly efficient polymeric biomaterials. Thanks. I acknowledge MNNIT Allahabad for providing me all the facilities. My collaborators, Professor Anuva Sharma from Punjab University, Chandigarh, Dr. Lokesh Kumar from Sardar Vallabhbhai Patel Agricultural University, Meerut, Dr. Uvanesh from Applied Mechanics Department, MNNIT Allahabad, and my sincere thanks to my senior, my brother Mandeep Singh, who created interest in my performing research. He mentored me, like I was telling, that we should mentor the students in such a way that they should generate interest in research. In the same way, he was the man who motivated me for coming into the field of research and academia and helping me throughout my PhD work. Thank you. Glimpse of the MNNIT Allahabad. Thanks for your patience. Any questions?
Hello?
Hello. Hello.
Hello.
Ask.
If the participants are interested, they can now interact with Dr. Ahuja. Hello.
I think everyone has gone for the lunch.
Okay, so nobody is
No,
interested.
no, one person has raised the hand.
If
Ashok
anybody is
Veera, sir.
interested, they can now discuss with Ahuja sir about if they have any questions or queries.
Hello. Hello, sir. Namaskar,
Yes,
sir.
sir. Go ahead.
Yes, sir. Sir, you have given a lot of information. Nice talk. But I'm having some question that in polymers, basically when you prepare in your PhD that nano hydroxyapatite, right? So if
Yes, yes.
someone is preparing, if you take that nano hydroxyapatite, and if he wants to load in a polymer, right?
Yeah.
So if he
Yes.
wants to functionalize, either you will prepare some bonds, or sometimes
Yeah.
people never used to prepare bonds, but they will load the drug loading of the drug. So my question is that if you load this hydroxyapatite in a polymer, okay, so how could you characterize that molecule using which technique? Either IR, is it via IR, or in which technique are you going
You
to
can use
characterize
IR
that
also. You
molecule?
can use NMR also, but we used IR. We use FTIR for characterization.
But in FTIR, you can see that that is not an organic molecule. How could I characterize this, sir? It's not an organic molecule. It's a inorganic molecule, calcium hydroxyapatite, I believe.
So IR cannot be used for characterizing inorganic molecules.
No, in organic it can be, but that hydroxyapatite, sir, there is no carbon, I think,
But
sir.
we are infusing it into the polymers, and mainly it will give the structure of the polymers. For the characterization of polymer, we can use FTIR, and for the characterization of ceramic, like hydroxyapatite, we can use XRD.
That mean I'm not
XRD,
understanding. Basically,
NMR.
I face a problem exactly, sir. That when I prepare, the polymer is having some FTIR characterized peak, sir, characteristic peak. But when I'm loading a drug, the drug is having a wavelength. Suppose 280 lambda max is there. So I know that lambda max is 280. In the polymer, that 280 is not there if it is not loading. But after loading, that polymer is showing lambda max at 280. But I'm not getting one.
Which drug you are loading?
Any molecule, I mean, the molecule which is having 280 lambda max, when I'm loading that one, sir, I'm getting that one. Suppose you take 5-fluorouracil drug, cancer drug. If you load, it will show you at 267 or near about 280 lambda max. So, but when you will do the IR, the after loading the drug will you see the peak of 5-fluorouracil? Because
If
it
you are
has been
not able
entrapped.
see, listen to me. If you are not able to do it using like IR, then you can use like when the polymer is in the viscous state, when before going into the like uh, stable form, take the sample from it, and you just put it into some organic solvent, and then you can go for UV, and for that you need to calibrate your initial drug, at what point it is showing
No, in
your
UV it's
calibration
okay.
curve.
Sir, I'm getting the data in UV. It's all right. But I'm interested to characterize using the FTIR, sir. I'm not getting that peak. Because people used to say that if the drug has been loaded, so in case of XRD, you'll see some peaks also. If you'll see in XRD, there is a peaks. But in FTIR, I'm not getting. But in UV, I'm getting the data. I'm not understanding why it is not happening.
No, most probably your reaction is not completed, or some other thing might be there.
Okay, fine. Okay, sir. Yeah, you have shown us a lot of polymers. Can you please tell us some polymer that is, I mean, having some fluorescence property? I mean, I will not take or targeting property. Suppose a molecule is there and a polymer has been loaded, a drug has been loaded in a polymer. And if I am interested to have a molecular imaging in a cell, I have to load some fluorescing agent, but I am not interested to load any further molecule in that. I'm interested that that polymer itself will show some imaging property. Is there any polymer? How you have shown us a lot of polymer. So is there any polymer which is having that fluorescence property, or like
some molecules are having that is your what is that light absorbing property also. If it can absorb the light also, so I can use from targeting point of view. Is there any polymer, because you are working in that area? So I'm
So
interested
actually,
to
I
know
am
this.
not working on
You have shown
fluorescent.
us a lot of
I
polymers.
am not working in the fluorescence area. But you can use like fullerene or some other thing you can explore. I am basically working on polymers for bone tissue engineering applications.
Sir, our next speaker is on the line. So we request the participants to send the queries to the FDP conveners and coordinators, and we will forward them to
Yes,
the
sir. I'll mail to the sir.
we'll forward them to the speakers, and you can get your
Yeah.
queries back. So as we are a time-bound event and we have to continue with our event, I would request
Sure.
the
You can use
Sure.
PTFE,
Thank you.
Teflon. You can use as a fluorescent polymer.
Okay,
PTFE,
thank you.
sir.
Thank you, participants, for your
Sir, I will ask
interaction.
you, sir. I will email you, sir. I will email
Yes,
you my
okay, sir.
question.
Sir, please send your question to the FDP conveners. Okay, sir?
So we are happy to have your question. You just
Yes.
send me the questions.
Okay,
We
okay.
will
You
let
can
you know.
send to Dr. Uvnesh, or you can send it to Dr. Gaurav, or you can send it to me also, Dr. Sumedha Mukherjee. Okay. So we will move forward with our sessions. We would like to thank Dr. Ahuja for this very clearly engaging session. We can see participants are very happy and they're very eager to participate. Your discussion on biomedical polymers had brought forward exciting perspectives and practical applications. We would request you to kindly accept a virtual memento in appreciation of your contribution to this FDP. I request Dr. Uvnesh Kashi Vishwanathan, convener from MNNIT for this FDP, to kindly present this memento to Dheeraj sir. Thank you.
Dheeraj sir, kindly accept the memento. It was very
Thank
engaging
you.
session.
Thank
Thanks
you.
for coming and gracing the occasion.
Thank you, Dr. Uvanesh, for providing this opportunity, and I'm also thankful to the whole of the team of this workshop. And thank you, Dr. Sumedha, for such a nice coordination.
Okay, fine. Dr. Gaurav, Saurav sir, you can start. Sumaida ma'am.
Thank you. We will move on to our next speaker. So our next speaker is Dr. Saurav Bhardwaj, who is a leading researcher from MNNIT Allahabad, who specializes in computational fluid dynamics and aerosol delivery modeling. Dr. Saurav Bhardwaj is an assistant professor in the Department of Applied Mechanics at Motilal Nehru National Institute of Technology, Allahabad. And prior to his current position, he was a postdoctoral scholar at the Artificial Heart and Cardiovascular Fluid Dynamics Lab at the Pennsylvania State University, USA, and a PBC postdoctoral fellow at the Faculty of Biomedical Engineering, Technion Israel Institute of Technology, Haifa. He also worked as a senior researcher in ESS engineering software Steer GmbH at Steer, Austria. His primary research interests include biofluid dynamics, cardiovascular and pulmonary fluid flows, and lattice-Boltzmann methods in multiphase, multi-component flows. He has published 22 peer-reviewed international journal articles in high-impact journals. In June 2016, he was nominated for In-Residence Program for Scholars of IITs, ISERs, and IICs B 2016 at Rashtrapati Bhavan, New Delhi. Research to the Honorable President of India, Shri Pranab Mukherjee, and Minister for Communications, Shri Ravi Shankar Prasad. He is also a recipient of prestigious the PVC Fellowship for Outstanding Chinese and Indian Postdoctoral Fellows from the Government of Israel in March 2020. He will today deliver a session on in silico optimization aerosol delivery in upper airways. Sir, we welcome you, and we are very eager to hear from your vast research experience. Thanking you. We request you to grace the stage. Thank you, sir.
Yeah, sure. Thank you. Good afternoon, everyone. So as Dr. Sumedha has given my introduction, I'll just briefly give some background that I'm not a biomedical engineer by profession or by my education or qualification. Basically, whatever research I have done in the field of biomedical engineering, it is based on the fluid dynamics fundamentals. So my background is in mechanical engineering, but using computational tools, you can go into any direction, right? And that is why today my topic of a talk is in silico optimization of targeted aerosol drug delivery in upper airways. So today I'm going to focus only on the pulmonary diseases. So to like in biomedical engineering applications, if you want to do any kind of research, you have primarily three tools, right? First one is in vivo, second is in silico, and the third is in vitro. Now, the problem with in vivo, if you go for in vivo technique, the problem is you have to do your live experiments directly on the patient. And it is sometimes very difficult to go for each and every factors which you wanted to analyze directly on the patient. So sometimes we go for in vivo studies when you go for the animal studies, but other studies we can do directly on the in vitro platform, where you basically create a biomedical chip or micro channel or something like that, and there you wanted to study whatever disease you want to go for. Just give you a brief, like if you want to go for asthmatic patient, you can create kind of airways, obstructed airways on your microchips or microchannels, and then you can see how the air is going to flow through that particular inflamed channel. Now, the problem with in vitro platforms are that it's very good, first of all, but the second problem is it is not reproducible, right? So if you want to go for different patient to different patients, you have to basically make specific microchannels for each kind of geometries. And you know that there is a number of variabilities present in the patient to patient geometries. So sometimes it is very difficult to make specific microchannel for one disease and then go for another microchannel for other disease. Now, to overcome this kind of drawbacks, we also have in silico. In silico is nothing but computational simulations, where you can basically create a geometry. Geometry is like CAD geometry, computer-aided designing geometry, and then you can use that geometry, which is based on the CT scans that you wanted to study for the airflow in the specifically in the pulmonary diseases. So which model to choose? It depends on you. What is your question? What question you want to address? If you know that what is your question, then you can easily choose your model. Sometimes we use in vivo, combination of in vivo in silico or in silico in vitro. Sometimes we go for in vivo in vitro as well. So if you talk about the respiratory tract, it is a very complex geometry, right? So it starts from your nasal passage, it has your oral passage, and then it goes to your larynx, then goes to the trachea, and then it goes to the terminal bronchi, ducts, alveolar ducts, alveolus, which is very, very small. So when you say lung, lung is not this only nasal passage or your trachea. The main lung is deep down to your chest, and which is very, very small in size. So if you look at the size, starting from the tracheal cavity to your alveolus, it goes from 2 centimeter to 100 micrometers. So you have to go for micrometer in sizes. So it is very complex to basically resolve the fluid dynamics or your fluid characteristics, starting from your nasal or oral cavity directly to your alveolus. So you have multi-scale model in that particular analysis. And because of that, if you go for the fluid dynamics, the fluid flow changes from laminar to like turbulent. So if you don't have fluid mechanics background, then I can say that laminar is a very slow moving fluid flow, which doesn't have any chaos. But when you transit from laminar to turbulence, turbulence is basically a flow which has chaos or eddies. So if you go, you have seen such kind of turbulence in river where you have a pole. Let's say there is a bridge and bridge has poles. So just before the pole, if the river is quite very small, like calmly moving, then just before the pillar, you can see that the fluid is very calm. So that kind of flow is laminar. And just after the pillar, if you see that there is some chaos, so that kind of chaos generated because of the turbulence characteristics. So even in this respiratory tract, starting from the trachea or oral mouth to your alveolus, you have variation from laminar to turbulence. And this variation is going to affect the drug deposition in your respiratory tract. Now, our target was to optimize the drug delivery, because you know that most of the local respiratory diseases, the first line of action or first line of drug therapy is the inhalation therapy. Inhalation therapy is what? In most of the houses nowadays you have PMDI, you have an inhaler, right? You also have nebulizers for your babies. These days every other house has nebulizer, right? And then you can also have, for adults, you can also have dry powder inhaler, right? So the problem with these kind of inhalation devices is that mostly particles are lost in the upper airways. What are the upper airways? Upper airways is basically combined with the oral cavity, nasal cavity, trachea, and like just above your chest. So that is known as basically upper airways. So most of the particles are going to deposit in the upper airways because of the impaction. Impaction is one of the characteristics, or so because of the impaction, like direct impact under the influence of the airway on the upper airways wall, so particles are tend to deposit in that area. But your ultimate target is to basically deposit your drug into your deep lung or deep alveolus, right? So one of the major drawbacks which we find in literature is that people are going to consider this vocal fold movement as a static, right? So you know that in our larynx area, we have two vocal folds. Now vocal folds open during the inhalation and it closes during the exhalation. These vocal folds are very, very important in phonation, like creating the speech, creating the sound. But in the drug delivery application also, this glottis motion is very, very important. What is glottis? Glottis is basically the area between these two vocal folds. So here in the animations, you can see in the top animation on the right, you can see the movement of the glottis, right? The glottis is opening, like vocal folds are expanding during the inhalation, and during the exhalation, it is going to contract, right? So this kind of motion we basically implemented in our CAD model, and then we have provided using the dynamic motion of the mesh. Now, what is mesh? Mesh is nothing but dividing the CAD geometry into small cells so that we can solve the numerical equation in each and every cell, right? Now, whatever literature you can see these days, like up to this point, what you have, people used to have a static kind of glottis structure, and they try to create a phantom, right? And then they basically analyze the airflow. But the problem with that is it is not transient motion. You can create one shape at certain instance of the glottis, and then you can see how the air is flowing or the particles are depositing. But that is not true when you go for realistic applications, because your glottis is moving as your time is increasing and decreasing, it depending on your inhalation and exhalation. So our idea was to see numerically how this glottis motion, like this transient glottis motion, is going to affect the particle deposition and airways. So what we have done, we basically collected the CT scans, patient-specific CT scans. We used some segmentation tools or softwares like Mimics, or you can also use TerraRecon. These are very advanced level semi-AI tools or softwares that can help you to segment the CAD geometry, computer-aided design geometry of the respiratory tract based on the patient-specific CT scan. So first task is to basically have the CAD geometry from the CT scans or MRIs, and then using this CAD geometry, you can basically analyze what kind of drug deposition you can have for different disease geometries. Now, as I previously mentioned, it is very helpful to go for in silico methods, because you don't need to destroy, you don't need to make and destroy the material in terms of microchannels, and you sometimes you have some complexity in the patient's treatment, and then you cannot do the direct experiments on that. That advantage we can have in silico methods. So in silico methods, you can go for patient specific scans, do the segmentation, create the geometry, and then do your simulations. But sometimes there is a question that how do you know that your simulations are giving the correct results? Now, to answer that question is that we need some kind of validation before starting the simulation. So there comes the combined model of or coupled model of in silico and in vitro. So what we do, we basically 3D print this CAD geometry, and then based on this CAD geometry, we basically we basically go for some in vitro analysis. So in vitro, to see the flow, we can go for PIV, right? PIV setup. So in every biomedical engineering lab, generally you have PIV. When you want to see the particle deposition, you have different methods. So for the in vitro particle deposition, you need to have an aerosol generator, or sometimes you can use readily available nebulizers or PMDI, pressurized meter dose inhaler, which we generally have for the inhalation, right? So these kind of, first you do the in vitro and in silico validation. Once you are satisfied with your results for the given geometry and for the given software schemes which you are going to apply based on the boundary conditions, then you can say that, okay, this method is successfully validated and you can go for further analysis based on your different diseases. Now, for our task, basically for the influence of the glottis motion, similar thing we have done. And as the geometry or the patient specific scans were based on the adult geometry, we selected dry powder inhaler as an inhaler tool. Now, on the left side here, you can see this is the dry powder inhaler, which we generally have. So what you do, you from this opening, you basically attach to your mouth, and then you try to suck the suck the particles based on your inhalation maneuver. So for the DPIs, you generally have this kind of breathing profile where your inhalation is very quick. Then you hold your breath for some time and then you release, right? This whole process is known as inhalation. Sucking the particles, holding, and then releasing, right? So what we do, we basically applied this breathing profile at the mouth inlet, and under the influence of this flow, we release the drug particles, right? To solve this problem, computationally, you have number of softwares like ANSYS Fluent, you have OpenFOAM, you have Console. It's up to you. Whichever software is available with you, you can go for that. So for this particular study, we selected ANSYS Fluent, and then we solved Navier-Stokes equation at each different cell of the mesh. Now, as I already mentioned that the flow transits from laminar to turbulent. So in this case, the Reynolds number, which is a parameter to select whether the flow is laminar or turbulent, is above 5,000. So that is basically in the range of turbulent regime. So we selected a model which is K omega SST. Those who have background in the computational fluid dynamics, they might know. K omega SST is a very good model to analyze the turbulence characteristics, although it is not top class model because there are different other models, but based on the computational facility and other stuff and your validation, you can select this model. Now, when you have a first, you basically have airflow characteristics throughout the respiratory tract model, then you basically select what kind of drug particle you want to use. Now, from the DPI, we generally have particle diameter in the range of 1 micron to 10 microns that you can generate easily. But the assumption which we have taken is that the particles, or in terms of drug liquid particles, that is aerosols, but this is the dry powder. So that's why we are saying particle. The particle diameter is in the range of 1 to 12 microns, and we consider that those particle shape is spherical, right? So generally, whatever aerosols or particles you generate from the DPI or different inhalers, they are in spherical shape, roughly. So that assumption is readily valid for the analysis which we have done. Now the question is, how do you judge that how much glottis will expand? So based on your previous in vivo studies available in the literature, you can see that for different age group of adults, how much glottis moves during the inhalation and how much glottis contracts during the exhalation. Although, as I already mentioned, patient to patient, things will vary, but average, if you take, you will get that the glottis area expands maximum 40% during the inhalation. So what we do, we basically have a central position of the glottis area, and then during the inhalation, we change the glottis motion from 15 to 40%. We wanted to see, or we wanted to analyze that, okay, 40% is the maximum. So we will see what happens during the 40% criteria, but we wanted to see that what happens in between. So we selected two percentages, which is 15% and 40%. And then we have done some kind of analysis, and we want, in that particular case, we selected few zones where we wanted to see the airway characteristics first. Second, under the influence of that airway characteristics, we saw what is the particle deposition at different sections, right? So as I already informed you, we used fluent ANSYS. Now different kind of mesh shapes or sizes are available that you have to create, and then you have to see for different number of mesh size, right, what is the independency on the results. So that is called grid independence study. So we have done different number of meshes study, like 10 million we have selected finally, and based on this 10 million cells, we have our boundary conditions. So at the mouth, as I already told you, we provided the flow rate condition, which is the DPI maneuver, and that flow rate we converted into the velocity inlet based on the mouth area or mouth diameter. And then all the walls we have selected as no slip. No slip is the condition where we assume that particles are going to stick at that particular interaction site, right? And at the outlet, we basically define weighted mass outflow, where you say that, okay, from the left side lobe, this much percentage of particles or flow is moving, and on the right side, top, left, different kinds of weighted mass outflow is passing through. Now, based on that, you can see some kind of mean velocity contours and mean velocity contours based on the different glottis area at different time instance. So on the left, you can see that we have shown the velocity characteristics or vector plots on the peak time. And that peak time of inhalation is known as peak inhalation flow rate. So when you have a maximum flow rate during the inhalation, what is the characteristics of the air inside your airway tract? So what we have seen that as you go from different sections, you can see that the flow intensity keeps on increasing, and that maximum you can have exactly at the notch of your glottis area. So this red color shows maximum value of the velocity, and that blue color shows the minimum value of the velocity, right? On the other hand, if you wanted to see only on the glottis section, you can see on the right side where we have plotted the average velocity, and you see that from time 0.1 to 0.25, slightly the velocity has increased. But as you go beyond this time instance, where you reach the peak inhalation flow rate and you surpass that particular value, you can see that the maximum intensity you will see and the area coverage is at the T equal to 0.45, which is nothing but your peak inhalation flow rate instance. And as you go beyond this time limit, you see that area coverage of the peak intensity keeps on decreasing. Now, if you want to see this particular change, unsteady velocity contours at various instant and sagittal plane and glottis section, you can see here. So at different time instance, we have also shown this kind of images, right? And this section now you can see exactly at the location where you have glottis area or your vocal folds. Now, based on that, we have also analyzed or investigated that what is the effect of this motion of the glottis on the recirculation zone. So because when you have a flow and some area is keeps on increasing and decreasing, the flow tends to create a recirculation zone. And when during the recirculation zone, when you inhale the particles, because those are very light in mass or sometimes it is massless, they try to move under this recirculation zone, and it is not basically pass through it because it doesn't have its inertia or strength. And because of that particular reason, the particle tends to deposit mostly on the glottis area region. So for the DGL equal to 1 is nothing but your neutral position of the glottis, where at T is equal to 0. This is 15% increase in the glottis area. This is your maximum, which is 40% increase in the glottis area. So at the neutral position, you see there is no recirculation zone. There is a slight, very minimal recirculation zone, which is not developed completely. When you go for 15%, you have two zones where one zone is very weak. And when you go for 40%, you see that, and mostly in the entire glottis region, you have three large recirculation zones, right? That is going to affect the drug deposition. And based on that, we can also calculate what is the strength of turbulent kinetic energy, because that is a turbulent flow. So you don't need to you have to consider this turbulence kinetic energy, how much strong turbulence you have during this motion of glottis area. Now, once you have this airway characteristics throughout your airway tract, you basically also see what is the particle deposition at different sections or different regions in your complete airway geometry. So here we have shown that for the one or neutral position of the glottis, this is the particle deposition map in the complete airway, whereas this is the maximum glottis motion contour map. Here you can see that mostly particles are deposited throughout the airway tract, but we have to focus more on this particular lower airways, right? This is called as bronchi. So why we are basically focusing on this particular area? Because whatever particles are passing through this outlets of the airways present in the geometry, that will basically reach to the deep alveoli or deep lung. And that is what we needed. So what we need less deposition in the upper tracts and more deposition in the lower tract. So we wanted to see in this case whether the glottis motion is going to affect or not. So based on the particle deposition on this airways, we can see easily from the deposition map or heat map, this red color zone or red color crystal cluster is nothing but the region where you have more particle deposition of that particular diameter. So here we have shown for 1 micrometer diameter and 10 micrometer diameter, and for a specific glottis expansion ratio. This is the neutral and 40% is the maximum one. And here you can easily see that for 1 micron particle, because the size is very small, so it has more tendency to pass through the airways, right? Pass through the glottis and then go to the deep like upper airways, which is these area. So here you can see that most difference you will see for 1 micron particles, whereas for 10 micron particles there is slight change, but it is not very much. So like visually you can see that there is some change when you consider the glottis motion, right? And not consider the static glottis. But what is that? Like quantitatively you have to mention, right? So based on the quantitative analysis, we tried to see what is the percentage change between the static glottis and the dynamic glottis, right? So what we have seen that for 1 micron particles, when you consider the static glottis assumption, when you consider that vocal folds is not at all moving, then the percentage deposition in the airways is about 6.24. Whereas when you go for your 40% increase, you will see that the particle deposition is about 16.73. So from 6 to 16.73, you have a drastic change in the particle deposition, because you have initially you have very low deposition, right? So this approximately 10% change is a huge amount in terms of drug particle deposition, right? So far, whatever assumption we have taken, the one of the most important assumption is your spherical particle shape, right? But when you want to deposit more drug into the deep lung, you need to have more payload in your particle or drug particle, right? If you consider the spherical shape particle and if you consider elongated elliptical shape particle, both have different capacity to deposit or to carry the payload. Payload is nothing but how much drug you are having in one particular tablet, or you can say in this case particle. So we also wanted to see, if you consider fiber shaped particle, fiber shaped elongated particle, what is the percentage of deposition in the bronchi region, right? How much particles and which range of aspect ratio particles are passing through the upper airways and going to the deep bronchi. So for that particular case, we have not gone for the CT scan particular. This is an idealistic geometry. We have created this geometry with deep lung or bronchi. And for the same DPI inspiration or inhalation, we basically go for analysis. And here we have seen that as you increase the aspect ratio, you have less deposition of higher aspect ratio particles in the upper airways. When you have less deposition of the higher aspect ratio particles in the upper airways, that means those particles are basically passing through this upper airway and going to the bronchi region, which is your ultimate target. So if you go in the bronchial prod, you can see that for certain kinds of diameter, right? If you go beyond 6 micrometers, you will see that in bronchi, higher aspect ratio will have higher deposition. So we are not saying that you always go for fibers. And the problem is you have to optimize. The good thing with the fiber shaped particles is that they are basically streamlined with the airflow, and it has more tendency to basically reach into the deep alveoli compared to the spherical shaped particles, and also because it has large surface area and it reaches deeper. So we say that always don't go for spherical shaped particles. We have an area where we can optimize the drug shape as well. Now this is very important. So you have seen that, like, for spherical shaped particles and for fiber shaped particles, you have different kinds of deposition characteristics. One of the major problem when you have local respiratory diseases like asthma or some COPD is that sometimes you need to have a long-term intubation just to have your breathing bypass. Now, when you have your long-term intubation, sometimes the patients get inflammation around this tubing, and that particular inflammation is known as subglottal stenosis. Now, when you have subglottal stenosis and you provide, if you remove the long-term intubated cannulas, the problem which happens is that it reoccurs again and again. Like I'm talking about stenosis. So it is not a permanent treatment of the stenosis. So we wanted to see what optimization or what treatment methodology we can use where we can directly deposit the drug exactly at the location where you have a stenosis or inflammation. So on the here you can see that these are the different glottis inflammation where you this white color cotton is nothing but it is starting of a cancer tumor. So, and this is subglottal stenosis. Here you can see that this hole is nothing but because there is a tube, there was a tube, and around the tube there is a formation of the mass lesion. So what we do, we basically have some tracheostomy at some initial tracheal rings, and then we insert a tracheostomy cannula. Now that cannula primarily is used for the breathing bypassing or phonation. Now we wanted to see whether we can use this cannula insertion for the particle deposition at the subglottal area or not. So our idea is to use fenestrated cannula. Fenestrated cannula has a hole somewhere here, and you can provide drug from this particular cannula opening. Here from the mouth, you can basically breathe normally if you have removed the your cannula. Otherwise, with the cannula only, you can have drug deposition as well as you can bypass the air. Now, on the right side video, we have shown, this is the PMDI PMDI pressurized meter dose inhaler. And then this inhaler is connected to the cannula opening, right? And when the idea is, generally we have this puff of drug during the inhalation. Rather than giving the inhalation drug puff, we ask our patients to basically have the puff during the exhalation. So when you exhale, there are more chances of having this drug movement towards the glottis side, and there are more chances of having your more deposition around the glottis or subglottal area where you primarily have inflammation. So this is a kind of optimization we are doing right now, where we wanted to basically optimize the drug deposition during the exhalation maneuver. And also we are basically developing a new class of cannula, right? So that cannula, the new class of cannula which we are developing right now, that will not only focusing on the upper side, that will focus on the lower side tumors or some kind of inflammation, right? So this is a new class of cannulas which we are going to develop, and this is a collaboration with the MDs, like doctors from the Israel, and they are also having some clinical studies. We are doing the in silico studies of this particular treatment. We have already published one paper, proof of concept, and the group from another Technion University, Israel, they are doing the in vitro. So this is a collaborative work, and we are basically focusing on that. So our initial task, whatever paper we have, in that particular case, we have inserted this kind of cannula. We created a geometry, right, which is our basic patient-based geometry, but it doesn't have any subglottal stenosis. As this was the proof of concept, we just wanted to see whether our inhalation and exhalation deposition of the drug, what we are presuming basically, is correct or not. So in this case, what we did, during the inhalation profile, like this inhalation profile, we tried to insert the drug during the cannula and from the mouth, and we just wanted to see whether the particle deposition increases around the glottis or subglottis area during the exhalation or not. So from this particular analysis, what we have seen is, if you focus only on the subglottal area, you can see that from the drug inhalation from the mouth, the particle deposition is just 0.119, and that is the fraction of the total injected particle. Whereas when you have the drug deposition from the cannula during the exhalation, you'll see that this particle deposition around subglottis area increased to 3.63%, which is a drastic change in the particle deposition during the exhalation close to the subglottis area. Other than that, some other regions where you can see that the particle deposition during the inhalation and from the mouth, particle deposition is more, whereas at the subglottal area you have more deposition during the exhalation and when you inject the particles from the cannula during the exhalation. So this is what we have done so far, and the conclusion I already told you, like glottis, we don't need to consider the glottis as static glottis, because it is not going to give you the correct results. This will give you the wrong results. You need to basically have some transient motion of the glottis if you want to basically optimize the drug deposition in different regions of the respiratory tract, right? You can also develop few methods where you wanted to target some specific disease like tumors at different locations of the airways, and then you can develop some kind of cannula designs. You can go for it. So far we have number of cannulas, fenestrated, without fenestrated, but the problem with that, no cannula is perfect. Different cannulas depend on your specific requirements clinically. So we wanted to develop a single cannula which can give you different options for different patients. So this is what we are doing right now. Thank you so much. I basically acknowledge everyone who is having some part or some contribution to my research journey so far. Thank you so much. If you have any question, you can ask. Hello.
Thank you. Thank you, Thank you, Bhadwaj sir, for a very highly technical yet understanding session. It has been a great pleasure hearing to you. Your insights have been of immense relevance of today's healthcare landscape. Please accept this virtual memento as a token of our gratitude. We'll not hold you for a longer time. We know that you have other responsibilities. So any participant having any query for sir can write to us, or they can put it in the chat box. We'll definitely forward it to sir. Thank you, sir.
Thank you.
Thank you. Thanks a lot for taking your time out. Thank you.
Thank you.
Our next speaker for today's session, for the final session today, will be delivered by Dr. Rishabh Singh from IIT Patna, a distinguished expert in the artificial intelligence applications in medicine and diagnostics. Dr. Rishabh Singh received his PhD in computer science and engineering from the Indian Institute of Technology ISM Dhanbad in 2017. He is currently serving as an assistant professor in the Department of Computer Science and Engineering at IIT Patna, India. With over 12 years of combined experience in industry research and academia, Dr. Singh has established a strong foundation in computational science. His research interests include image processing, signal processing, and deep learning models with a focus on developing intelligent systems and real-world applications. He has published more than 34 research articles in reputed international journals and conferences. Dr. Singh is also an active member of the prestigious professional bodies, including the IEEE and the Association for Computing Machinery. He will be today speaking on the topic of AI in healthcare. So we are honored to have you with us today. Please proceed with your session. The stage is all yours. Thank you.
Dr. Sumedha, is my voice audible?
Yes, sir. Your voice is audible, sir.
Okay.
Sir, voice is audible.
Sir,
Yeah.
would you please turn on the camera?
Camera, it's shared. Just give me a second.
Okay, sir.
Camera is already on.
You are not visible, sir.
One second. Yeah, it's visible now. Camera.
Yes, sir.
You are visible, sir.
Okay. Shall we start?
So the stage is yours.
Okay, thank you so much. So today I will be going through the artificial intelligence in healthcare. Although over a decade, people have contributed a lot in this area, and there has been so many research articles which has been published, patent, and even the product have came up in the market. But I'll try to take this in a different dimension altogether. Like whenever we are talking about artificial intelligence, we are taking into the term deep learning and machine learning, and we know that for these kind of framework and technologies, uh, we need a huge amount of data. But the problem with the healthcare is, in healthcare there are diseases which are labeled or annotated as rarest of the rare disease. For example, if you take a survey, let's say randomly, out of 2 lakh people, hardly 2 or 3 people will be having cancer. And that cancer of which category it is, that is another issue. So, first of all, the data set which we require in case of healthcare, it's very difficult to get a balanced data set. Second, point is the disease which is there, if you try to subcategorize, it becomes another difficult task. When we talk about traditional machine learning versus deep learning, I'll first take you to the concepts why it is important, like more data is important, and how now the paradigms if in deep learning is shifting, and how it is shifting. So when we talk about machine learning, So you have giving an input to some feature extractors, which we call as feature engineering over here, you can see. And later on, that feature extraction and all, once it is there, then it goes to the classification model, and finally it gives the output. That means in machine learning, the classification part and the feature engineering part, they both needs to be separated, which was initially there. Now it was becoming very difficult to identify that which features are important and through handcrafted features. So what deep learning did is they clubbed it together. You send a raw data, the system itself will try to figure out and extract the features, and similarly it will go with the classification thing and give you the output. What you see over here is these neurons, the circles, these are termed as neurons, and these are your having activation functions which introduce non-linearity. Later on there is weights, so these edges which will contain some weights. So initially the features are sent, you reach over here and then you back propagate. So if you try to go with this, you need to have different samples of the data. There should be variations in the data. That's why people came up with augmentation techniques to increase the number of samples, because they need to bring the variations, otherwise the system will not be able to understand or will not be able to capture uh, the patterns which are there. It will be very rigid to the data which is given to it. It will not work with the, what you can say, the variations. So when this deep learning revolution turned up, where you need huge amount of data and you need very high computational server, so your data has to be huge. Most of the paper, if you see big papers which are published, which are highly cited, you will find that people are using data set known as ImageNet. So the ImageNet data set contains more than terabytes of data, more than 1,000 classes, none of them which is having medical images. And in order to execute such a huge data set and apply machine learning or deep learning algorithm, you need to have a huge GPU facility. You need to have very high computational power. So the question is, transfer learning, can it solve it? Now, what is a transfer learning? Transfer learning is, if I tell you like this, let's say you have a 4 year or 5 year kid. You take that kid to the IIT Patna, and let's say you said that this is a computer science department. People here work on computers, they generate algorithms, they work on hardware, and you explain everything related to the computer science department. You take that kid to another institute, let's say NIT Delhi, or let's say Galgotia University, wherever you take them, and you just have to tell the kid that, okay, this is a computer science department of Galgotia University, this is a computer science department of NIT Delhi. You do not have to give the kid the entire, what you can say, the description of the what the department does. You don't have to explain him that people again work over here in the field of computer science and they work on computers. So that means a knowledge gain, the student which gained, the kid which gained the knowledge at IIT Patna, he has retained that information. The information is still with the kid. He doesn't have to regenerate those informations. Now, when we are going with this transfer learning, how the question arises in the kids, okay, the kid has learned and he has used it, how we can make the models to learn and then transfer that. So first of all, the question arises is there are three basic questions which has to be answered: what to transfer, when to transfer, and how to transfer. So when they talk about these three questions, the first one is what to transfer. For example, I have built a model, a deep learning model, and I am just transferring the framework That means how many neurons will be there, what is the architecture, but I am not sharing the weights of the model. I'm not sharing the detailed parameters. Or I can share the architecture and parameters both. That means I need to ensure that which particular section is required for transferring the knowledge. The question arises is when to transfer. When we say when to transfer, the most important thing is first we need to understand the data. If your data is highly imbalanced, then we need to go with the transfer learning. That's the time. Or let's say where you have very limited infrastructure for high computational thing, there you can go with transfer learning. Or where data is very less, there you can go with transfer learning. That means if the data is imbalanced, which is the case of medical images, the data are highly imbalanced, or I would say the data is having what I can say, small amount of data or small amount of samples present in each class. The third question which comes to is how to transfer. So transferring basically happens is you take a model, you execute that model, you tune that model in a huge data set, let's say ImageNet. Now you will be using that model and its architecture to transfer it to your own data. For example, let me just show it over here. You see over here, these are some pre-trained models, exception, VGG16, VGG19, and there are lots of parameters. You just see the size of the architecture and you see the parameters. So if I want only the architecture or I want the architecture and parameter both I can select. Then you see over here is accuracy. You see the accuracy is over here. So the latest one is Next Large, which is having 350 million parameters. You just imagine 350 million parameters which you have to fine tune. So what I can do is I can take this model and use it for my application. That means there is a model we already have whose accuracy we know this is top 1 accuracy, this is top 5 accuracy. We know that. And now I want to use that in our own system. So let me show you one more paper. Why I am saying it is important. You see the distribution of the class. These are classes, ductal carcinoma and all. Adenosis, you see there are just 113 samples and this is 903 samples of ductal carcinoma. That means you see the data set is highly imbalanced. You see these are the three data sets which has having very low information about the data of their classes. Each class has just 113 samples and over here this class has 903. That means the data is highly imbalanced. Let's say move over here. You see this is another data set. Yeah, this is I think is the ISIC data set. Yeah, ISIC data set, that is the skin license sections. You see there is one class which has 6,705 samples. There's another class which just has 115 samples. That means out of 1, 2, 3, 4, 5, 6, 7, out of 7 classes, there is a class which is extremely having huge number of data. Then the second one is 1000 around, then 500, then 300, then 100. So the distribution of the data is very what you can say irregular, and the number of samples in each class is very less. So when I said when to transfer, that means there is a need when the data is highly imbalanced. Otherwise, what will happen is the model will have more influence of this particular class, or the model will be biased towards this class, which we don't want. So that means there is a, in medical images, there is a high chance that your data will be highly imbalanced. If you have any questions, any queries, you can stop me and you can post your questions over here. If you have any doubts or any queries. Okay, these are no questions. So we'll proceed. Now you just see this. How do we go about this transfer learning? So situation where what has been learned in one setting is exploited to improve generalization in another setting. So you have task A and you have task B. You see over here, the table is a square table, or I would say the field is square. There are two players playing. There are ball over there and so on. That means if a person has learned something about this particular game, table tennis, he will not have the complete knowledge of long table tennis, but yes, up to some extent he will be having the knowledge of the game, how the ball is moving, how the points are being calculated. If you miss, what are the rules for giving the service and all. That means I can learn some of the parameters over here and I can use it over here. I do not have to relearn that again and again and again. So these are some of the samples of the pre-trained models which are there. Now, what are the advantages of transfer learning? So advantages of transfer learning. So if you see over here, this usually the transfer learning has higher slope. This is the training, the epoch I can say, and this is the performance, the accuracy you can measure on that. So you see, initially only there is a higher start. That means the model, when it starts learning at initial stage, it has gained this particular amount of information. He already has. Then the second thing is, when you are gaining the knowledge, when the accuracy is improving, then again there is a steep slope. The slope is very high over here, and the finally also it's there. That means when you're transferring the knowledge, initially the person has some knowledge, and over a period of time, as compared to any raw model, this will give you a better performance. But again, that question, when to transfer, that is very important. So I'll take you to the first case study where the imbalanced breast cancer classification we have done using transfer learning, and this was published in 2020 IEEE SIM transaction of computer and biology and bioinformatics. Now, when we talk about the breast cancers, uh, what happens is it's currently in India approximately 80%, what I can say, of breast cancer cases are there. Just give me a second, let me just switch off this. 80% breast cancers are there, and this is one of the top leading or the main cause of having deaths in women, although it's rated as the second leading thing, but still that's a very huge number. The problem with the what I can say, the traditional method right now is it's improves the survival, but the thing is you need to have an expert pathologist, and that pathologist, whatever he does, is actually time consuming. You cannot just get the values, or you just not get the results in a short period of time. It's actually time consuming. Then you have histopathological diagnosis. There are H&E strains which are there, which is important for this to get the result. Then there are manual analysis, which suffers and which has variations related to the, what you can say, the inter observer or intra observer thing. But the question is, like, how to speed up the process. Speed up the process means how I can go around and I can get the results at a very early age, or if the diagnosis can help us to get the result at a very, what you can say, reasonable amount of time. So, if I talk about histopathological diagnosis, so to confirm IDCs, doctors actually rely on histopathological examinations of biopsy samples, and these samples are actually used in, what you can say, the hematoxylin and eosin, which commonly referred to the H&E staining, and then analyzed under the microscope. So if I'm saying, it's actually unfortunate, because the manual analysis is prone to variability between the observers, which I said, the inter observers. So there is a challenge in automated classification because of this. So despite these advancements and automated classification, for the cancer thing, they faces several challenges. The first one is the data sets used in histopathology are often highly imbalanced, meaning there are far more IDC images than non-IDC images. I have already shown you that example where we just had, in one class we just had 115 images. Second thing is convolutional neural network, which are commonly used for image classification, requires large amount of annotated data, and this is actually difficult to obtain in medical field, where labeling requires expertise knowledge, and at the same time it is highly time consuming. So if I conclude, the training these deep learning models are computationally intensive, and the limited availability of the labeled medical images, which further actually complicates the task. Now, if I see the solution of transfer learning, so now that we have seen the challenges of automated classification over here, now of the cancers, and more specifically the breast cancers, we need to discuss something of transfer learning which comes as a practical solution. So by the definition, if you go, transfer learning is a technique where a model is trained on one domain and is adapted to perform tasks in another domain. And in this particular paper, or in this study, I would say the the work which we did, we actually trained the model, which we actually used the model, which was trained on ImageNet dataset, which is a large collection which I have shown you recently. And they then applied this learned knowledge, the knowledge which they have used while training it on the ImageNet, VGG 16 and VGG 19. We went with VGG 19. We used that VGG 19 model, and we just extracted the features from the VGG 19 model. We did not retrain the model. So this approach has some advantages. First one is it significantly reduced the training time, since the model already understands basic image features. And secondly, it helps mitigate the issue of class imbalance, which I have already shown you. In this work, the data which we used was IDC breast cancer data, invasive carcinoma, ductal carcinoma. But the thing is, uh, the data which we had, it had around 2,75,000 or 2,80,000 images. Out of 2,80,000 images, 1,75,000 images belongs to one class and other class just has 80 to 90,000 images. So the imbalance was just the double. So to overcome this, we use the VGG 19 model. The term 19 and 16 refers to that there are 19 trainable layers. If you see the model, there will be more than 19 layers, but out of which 5 layers actually they do not train, like the max pool layer. They do not train anything, they do not update the weights. Okay, so 19 refers to the number of layers which is there. So the VGG19 actually extract the feature from the histopathological images, which are then used for classifying IDC versus non-IDC tissues. Now, if we talk about the motivation, so the first one is the limited availability of the labeled medical data. So the medical data sets, especially those involving histopathological images, are often small and difficult to obtain, like general images. And this medical images requires the expert annotations, typically by the trained pathologist, which I said and we have discussed, that is actually time consuming and expensive. So this scarcity of label data makes it a challenging to train deep learning model from scratch. Transfer learning helps overcome the limitation by allowing models to start with pre-learned features from large data sets like ImageNet, reducing the need for extensive labeled medical images. High cost of annotation. So if I talk about this, the labeling of medical images, like we already discussed, demand domain expertise. But unlike labeling everyday object in general image data sets and identifying cancerous tissue in pathological images requires detailed knowledge. And this makes the annotation process costly and slow, because the transfer learning reduces the dependency on large annotated data set by using pre-trained models that already understand the basic visual patterns, thereby minimizing the need of exhaustive manual labeling. What you can say in the target domain. Next comes the complexity. See, histopathological images of breast tissues are what you can say are rich in detail and contain complex structure that are difficult to interpret. And these images often include subtle variations in texture, color, and shape that distinguish healthy tissue from cancerous regions. And training a model to recognize these patterns from scratch requires a large amount of data and computational power. So the transfer learning provides what you can say a head start by using models that have already learned to identify and general image features, making it easier to adapt to the complexity of the medical images. Now, in many medical data sets, especially those related to cancer, there is a significant imbalance between classes. So there may be far more images of non-cancerous tissues than the cancerous one. Though the imbalance can lead to biased models that perform poorly or minority classes. And transfer learning helps mitigate this issue by what you can say the starting with the robust feature extractor that that is less sensitive to data imbalance, And this actually improves the model ability to detect rare but critical cases like IDC in the case. So when we talk about the computation, we have already discussed it reduces the transfer learning. And if the model is already trained, we can get good results at an initial layers. And another thing is the generalization, which is most important, and because this forms the basis for the federated learning. So the generalization is models trained via transfer learning tend to generalize better to new unseen data. And this is particularly important in medical applications where diagnostic tools must perform reliably across diverse patient populations and imaging conditions by starting with the model that has already learned to recognize a wide range of visual features. And transfer learning enhances the robustness and adaptability of medical image classifiers, leading to more accurate and consistent results. So, the breast cancer, if I talk about particularly IDC, remains a major health concern globally. Accurate and early, what I can say, diagnosis is critical. Yet, manual histopathological analysis is time consuming and prone to vulnerability. So, this study proposes a deep learning based framework using transfer learning to automate and enhance IDC classification, especially in the context of imbalanced data sets. Taking class imbalance in medical images or tackling these issues. If you see over here, this is IDC 0 and IDC 1. Just give me a second. Just give me a second. Okay. So these are some of the samples which are there. Now, if I talk about the contribution in this work, the first one is based breast cancers, particularly IDC. It remains a major health concern globally, and the early diagnosis is critical. So, one of the core challenges is the medical image classification in the imbalance data, which is the imbalance in the data set, where cancer sample often outnumber or the healthy ones. So this imbalance can skew model performance, and this paper actually directly addresses this by designing a framework that maintains a high accuracy and sensitivity even when trained on such skewed data distributions. Now the question is leveraging transfer learning with V knowledge. So the first one is it reduced the training time, the benefits from the generalized feature extractions, and adapt effectively to target domain of histopathological images. Now, third one is large scale data set utilizations. See, unlike many prior studies that uses subsets, this research employs the entire data set of 277 images, 1000 images. And what we did is we tried to compare this with multiple classifiers, the random after extracting the features using transfer learning. We applied the classifiers like random forest, support vector machine, logistic regression, and fully connected deep layers. Now, if I talk about the negative transfer, which is most important, sometimes what happens is when I am trying to make a model learn about something, suddenly what happens is the model start learning negative features. And when it starts learning negative features, the classification or the accuracy or the performance of the model will reduce. So this negative transfer needs to be tackled, and although this work did not get the results of negative transfer, or not much work has been done, but it just gave a direction. Now the question is, how does it work? If you see over here, this is my input which is coming to the model, and where feature extraction is done using VGG19. So this is CNN1, CNN2, this is max pool. These two are convolutional, then again max pool. Then these four are convolutional, then max pool, like that. So we have block 1, block 2, block 3, block 4, block 5, and finally you have fully connected layer. So when you are once you are sending the images over here, what happens is this image and this convolutional, which has some weights, they will perform convolutional operation. After performing the convolutional operation, the feature which is there, it gets transferred over here to the next layer. Now, these models, when they are designed, the initial layers which are there, they are giving you more generalized edges. Now, you just imagine this way. When a kid is born, he first starts identifying the edges. Whenever you see something, there's an edge. You see there's this line. Why we are able to see this line? Because there's a color difference, black and white. That's why we call that as an edge. Any place where there's a huge color difference, we are calling it as an edge. So initial layers, what happens is they try to give you generalized edges. And the last layers, they are more specific to the data set. The model or the data on which it is being learned, it is more specific to that. So what we did is, from each layer, we extracted the features. We did the convolutional operation and got the result. So let's say I'm talking about this. That means the image went with convolutional layer of this one, and this one. And after doing the convolutional of this, then whatever the feature is getting generated, that I am utilizing. So in normal, what you can say, the model of deep learning model, when we are sending the data forward, we have convolutional layers, and then we have fully connected layer. After that, we have the softmax layer, which gives you the classification of the based on the probability. But what is the thing over here? The most important thing is this fully connected layer. Now what we did is we removed this softmax layer and fully connected layer completely. We just focused on this convolutional layers. From each layer, we extracted the features and we saved it. So you see over here, we completely removed this particular section. We fully connected and the softmax layer, we completely removed that. Now, this transfer learning using VGG19, when we are sending the images, so we have block 1, block 2, block 3, block 4, block 5, then we have classifier, IDC0, IDC1, that are the classes. So these lines which you are seeing, that means it is extracting the features. So from each layer, we are getting the features. So first what we did, we completed the block 1, we got the feature, we trained the classifier, we got the result. Now this classifier can be anything. Either we can go with the fully connected layer, or we can go with the one which you call that as a random forest, KNN, anything. So let's have a look at the numbers of extracted features. So number of features extracted from different layers of VGG19. So if you see over here, the VGG19, you have block 1 con1, which is 1,60,000 extracted feature. Block 1 conf2, that also has 1,60,000. Block 1 pool has 40,000 features. Block 2 Convener has 80,000 features. So you see how many features has been extracted from each layer. So after extracting the features from each layer, what we are doing is we are sending this to the model for the purpose of classification. Now what we did, we send it with the logistic regression, we send it with the SVM, random forest, dense layer, and retraining with the dense layer. Once we have got this, once we have achieved this, you see the precision 94.21, which is higher than the dense layer. Precision is very higher. Other parameters, there is mix and match. But the most important thing is when we are having transfer learning, your accuracy is improving or is almost similar to it, but the computational cost, that the learning time has reduced a lot. There is a lot more than the things which have come down. So if I want to conclude the work which is there, that means I do not need to retrain or do not need to train the model again and again. I can use some model which is trained on some other data set and use it over here. You just imagine this thing like whenever you are having some problem, you go to your seniors. They might not be the expert in the area, but they might have the wisdom to give you a path, to give you the way. Same way, in case of transfer learning, the models which are there is they might not give you able to give you direct solutions, but they will help you to overcome that because they will provide some additional information which will help it to get the results. Till now, any doubts, any questions? Okay. Now the question arises is, if transfer learning is there, then and it is actually reducing the performance, the reducing the error. So why can't we directly go with this? See, the transfer learning and federated learning, they both are designed to address the key challenge in the domain of learning algorithms, particularly in the case of healthcare where data is scarce and it's sensitive and it's often soiled. So both approaches aim to improve model performance with what you can say with limited label data or reduce training time and computational cost. Now this scalable AI solution that can generalize across diverse data set. In medical imaging, especially for the breast cancer, these goals are vital. Annotated histopathological images are expensive to obtain, and patient privacy in case of breast cancer must be strictly protected. Both the learning paradigms often pathways to build robust models under these constraints. So that means I need to have something where the organization shall not share the data so that it can retain its what you can call that it's protect its privacy, I would say, and at the same time they are coming up with a generalized model. So if I talk about the limitation of transfer learning, is first one is the transfer learning typically requires access to a large centralized data set for pre-training, which is not feasible in medical domain. Like when we talk about VGG19, VGG16, all those were trained on huge medical domain. Then there are some privacy concerns. Medical data is sensitive, and centralizing patient data from multiple hospitals for training can violate privacy regulations like the GDPR and all. Then the hospitals and labs actually often store data locally, and they are reluctant, or I would say they are legally restricted from sharing it. These pre-trained models may not fully capture the essence of medical images, especially when trained on general purpose data set. Like we saw, there were some classes which are having just 113 classes, 113 samples. So there is a need of federated learning. Federated learning is actually a decentralized machine learning approach where multiple clients, example hospitals, they collaboratively train a shared model without exchanging raw data. Like each client will train the model locally on its own, since only the model updates to central server. The server then aggregate these updates to improve the global model. Now, this process is repeated iteratively, allowing the model to learn from distributed data while preserving privacy. Now, the thing is, the question arises is why federated learning is needed in healthcare. Now, federated learning actually ensures the patient data never leaves the local institution, aligning with the privacy laws and ethical standards. So, the cross institutional collaboration, let's say there is a hospital A, there's hospital B, there's hospital C, they all want to have they all want to have a model which can classify their data, but each one of them individually, if I talk about, they have very less samples. But if there is a way where each of the organizations can ABC hospitals can hold their data, they do not share the data, but instead they share a model. So there is a model which is trained on A, there is a model which is trained on B, there is a model which is trained on C. Once these models are trained, after training the model, we share the values. Okay. Now, the thing is, this work of federated learning actually learns from a wide variety of patient demographics, imaging equipment, and clinical settings. And this actually improves the generalization or generalizability of the model. So the institution retains control over their data and increasing trust in collaborative AI development. So in the context of breast cancer diagnosis, federated learning allows multiple hospitals to build a powerful diagnostic model without ever sharing sensitive histopathological images. Now, if I talk about transfer learning and federated learning, they are not mutually exclusive. They can be combined for a better and greater impact. So, the local transfer learning, where each hospital can use a pre trained model like VGG19 and fine tune its own data using transfer learning. So model A, model B, model C. They are trained on dataset A, B, and C belonging to respective organizations. Once they have trained, what they can do is they can do a aggregation. They can do an aggregation somewhere and then use the aggregated model for the purpose of training. So these locally fine-tuned models actually get aggregated, and this is known as federated learning, to create a global model that benefits all the organizations. So the hybrid approach leverages the strength of both paradigm, transfer learning and federated learning. Now, when we combine both the approaches, it's actually local fine tuning ensures the models to adapt specific data distribution. And this pre-trained models actually reduces the number of what you can say the training rounds needed in federated learning. Now, there is something as privacy preserving personalization. So, each institute can maintain a personalized model while contributing to a global one. This synergy is particularly useful in medical imaging, where data is both sensitive and heterogeneous. Now, real world relevance and strategic shift. If we talk about, so imagine a network of hospitals working together to improve breast cancer detection, and each hospital actually trains a model on its own histopathological images using transfer learning. So these models are then federated to build a global diagnostic tool, and the result they result a highly accurate and what you can say privacy preserving system that benefits from diverse data without compromising patient. Now, strategic shift means earlier from local system people move towards a distributed system. In distributed system, people initially worked with the centralized way of having a global model. Now people are shifting towards a decentralized architecture. That means a distributed environment with a decentralization. Now, if you look over here about the federated learning, if you look over here about the federated learning, you just imagine db1, db2, db3, db4. So all these are the considered as different hospitals, and each hospital, you see there is a learning system which is there. So this learning system learns on database 1, another learns on database 2, this learns on database 3, and this learn on database 4. So each one of them, this learning system learns locally. And once this process is done, each one of them have learned locally. I'm going to combine these learning systems and create a global learning system. And that is known as federated learning. So how do we start? So there is a central server, which is over here. It sends a architecture. It does not share the data. There's no shifting of data. It's just share a structure, an architecture. That architecture reaches each of the organization. Now, this architecture, the weights are random. At the first stage, each architecture of different organization will have the same weights after initializing the weights. Now, this DB1, it will train this model, DB2 will train this model, and DB3 will train this model. It will fine tune it and optimize the weights according to its data set. Once the data set has been used to optimize the model, these models and then sent back to the centralized server. Now, once these three models, or more specifically, the weights of the model, once it has reaches, because the structure is already there. Now, what it will do is, now there are different algorithms. Some people have used fed average. That means they will aggregate the weights. That is known as fed average, federated average, you can say. And once the average is done, now they will send it back, the weights over here. Again, the system DB will try to optimize this by its own data. So this process of shifting from here to here, here to here, again it will take lots of time. Means lots of time, but lots of epochs. so that there is a way where almost all the models will remain at the same level, and this is known as federated learning. So this model which is there, this can be a pre-trained model where it's a transfer learning and which is coming inside the federated learning. Till now, any doubts, any questions?
So please go ahead.
Okay. So first one is there's no data sharing and there's low network overhead. Low network overhead because we are not sharing the data. We are actually sharing the model and its weight. We are not sharing anything else. Now, if you see, one second. Yeah. If you see the federated learning, we actually use in MedSec Fed. There's another paper which we published in the year 2021. So in recent years, actually, the world has witnessed an explosion in the volume of medical data that are getting generated by hospitals, insurance providers, and patients. And this includes electronic health record, diagnostic images, lab results, and wearable device data. Such data holds immense potential for AI driven healthcare, enabling early diagnosis, personalized treatment, and efficient resource management. Now, deep learning in particular has what you can say shown some remarkable success in medical images analysis and detecting diseases like pneumonia, cancer, or COVID 19, or X rays and or MRI. So, however, these models requires large, diverse, and well annotated data sets to perform reliably. Now, the challenges with the centralized learning, despite the promise, the centralized deep learning is impractical in healthcare. See, medical data is fragmented across institutions, private and sensitive, governed by strict regulations, and difficult to share. So, aggregating data from multiple hospitals to a central server is not only a risky, but it's oftenly prohibited. So, federated learning actually offers a solution by enabling collaborative model training without sharing raw data. And each institution trains locally and share updates with the central server. But the thing is, however, traditional federated learning methods like Fed average, which was the first paper, they come up with their own set of problems. They share model parameters which can be exploited via model inversion attacks. And they are vulnerable to model poisoning, where malicious clients actually corrupt the training process. They suffer from communication overhead, exchanging large models every round. And they assume homogeneous client architecture, which is unrealistic in diverse medical settings. So, looking at this limitation of Fed average in federated learning, we need a secure and privacy preserving with no model parameter sharing. That means we are not going to share any model parameter with the central server. Robust to adversarial clients, maintaining performance in hostile environments, communication efficient, suitable to low, what I can say, the low bandwidth networks. They are flexible, diverse clients are there. So actually, these were the motivation behind this particular paper, where we needed a framework that can combine data-free knowledge distillation with model inversion to enable secure, scalable, and collaborative learning. Now, if we look about the motivation and we discuss about the motivation of this paper, actually in healthcare, the data is both highly segmented and fragmented. Okay, it's sensitive and fragmented, and patient records, diagnostic images, and clinical notes are distributed across hospitals, labs, and devices. Now, this fragmentation actually creates a barrier to building a robust and generalizable AI model. Now, federated learning emerged as a promising solution. It allows institutions to collaboratively train model while keeping data local. However, this traditional federated learning method like Fed average still have limitations. They share model parameters with central server. These parameters can be exploited through model inversion attacks, potentially leaking sensitive information. And federated learning is also vulnerable to model poisoning where malicious clients corrupt the training process. See, knowledge distillation is another technique used to compress models and transfer knowledge, but KD typically requires large and labeled dataset, something that's hard to obtain in the medical domain. For example, let's say annotating chest x-rays requires expert radiologist. And for emerging diseases like COVID-19, labeled data may be scarce or unavailable altogether. Now, challenges with existing federated learning method. If we talk about the they are not robust in hostile environments with adversarial clients, and their communication heavy requires megabytes of data exchange per round, and they assume homogeneous client architecture which is unrealistic in real world deployments where devices vary in capability. So the need for a new framework, so what we did is we went with this model but we didn't use the fed average. Now, if I talk about the key contributions of this particular paper, the one which we have, uh, in this, the most innovative aspect was data free knowledge distillation. So instead of relying on real labeled medical data, which is often hard to obtain due to privacy regulations, we use model inversion to generate synthetic medical images. This means we can train models collaboratively across institutions without ever sharing real patient data. And it's a breakthrough in privacy preserving learning, especially for domains where data is scarce or highly regulated. Then, uh, see, in whenever we are talking about federated learning in real world, not all participants can be trusted. Some may be compromised or adversarial. So this paper was tested in an environment where 20 to 40% clients were malicious. That means we were injecting noise or flipping the labels. That means class A was having a label of class B or class C was having a label of class A. So that is a flipping of labels we did. And despite these challenges, MedSec Fed maintained strong performance and even outperformed Fed average by 15% in hostile environments. This robustness is essential for deploying federated systems in open or semi trusted networks. No model sharing parameters. See, unlike traditional federated learning, this paper does not share any model weights or gradients. Instead, it's just exchange the logits, which are less informative and much safer. And this design actually prevents model inversion attack, where adversaries try to reconstruct training data. Just give me a second. हाँ जी पहुंच गए क्या हाँ 3:30 3:30 ठीक 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 मैं बोल दे रहा हूँ Yeah. So we were talking about, no parameter sharing. So we were just sharing the logits. So the design prevents model inversion attack, and because we are not sharing any parameters, the file size is less, and that's why it's more computational efficient. Since we are running out of time, this we have already said. Let me just explain you the model what we did. First one is we have some random noises, and using this architecture we got some updates, and there is a data which has been generated using model inversion. So this data set B is distributed across all the clients. All the clients has model B, data set B. Now this client 1 is trained on its own data set, and a data set which is generated using inversion attack. Using both the things, the model is trained. Not just only its own data, but a data which is common to all the clients that has been used. So once they have trained this average logit, which you see, it goes over here. There's a central server which aggregates the logits from each client and then send it back to the client. So we are not sharing the architecture. The architecture, all the clients can have different architecture, which is earlier, which was there. They can have different weights and they can have different data sets. So there is nothing which is common. Only common is the data set B, which we have shared with them. So since Fed average communicated around 44 MB per round, this was in KBs per round when we started. So if you talk about this is ideal for resource constrained devices and slow network environments, and enables scalable and practical deployment in real world medical settings. Okay. Now, if you want the work which is there, that it improves diversity of synthetic data for knowledge distillation, it enhances the quality and variability of generated images, and it actually the generalization is the most important aspect. Now, this thing applied this MedSec which we have done on the X-rays, chest X-rays, it can be taken forward to MRI and CT scan or the histopathological image. We have not done that. Third point, we have shown you the privacy preserving techniques, but it can still be ensured, or it can be still be secured using blockchain for metadata sharing for the logit. It can be used, but I'm not very sure on how it will go. So what we discuss is about the machine learning, deep learning, machine learning versus transfer learning, and the federated learning. So this is what's right now in the healthcare of AI. If you have any doubts, any questions, you can ask me. If you have any questions, any doubts, please ask me.
Participants are requested to interact with the resource person. If you have any doubt or any questions, you have any topic that you would like to discuss, sir is here. You all can interact with sir. Participants can now unmute themselves and can interact with Dr. Rishabh.
Hello.
Yes, sir.
Good afternoon, sir. It's a very nice presentation.
Thank you so much, sir.
Sir, can you provide that article, your triple E transactions that you have present? This model.
Transfer learning, yeah, it's there. I'll just share it.
Yes, sir.
I just shared it in the group.
Okay, sir. Thank you, sir.
Thank you.
Any further queries from any other participants?
Hello.
Yes, sir.
Sir, very good afternoon, sir. For example, we want to design biosensors.
Sorry, ma'am, your voice is not audible very properly. Please repeat.
For example, we want to design in
Biosensors.
yeah. So how we can how we can
Biosensors, when you're talking about which type of biosensors, the FBG biosensors or what? Because see, there
which
are some
detect disease,
areas
sir.
where we have worked, and in that we have figured it out which is giving us the highest figure of merit. And it was a
Okay.
combination which we are trying to optimize for the thickness and the wavelength. So you're talking about
Okay.
that kind of biosensors?
Yes, sir. Yes, sir.
Yeah. So
Means
for
we
that,
being we being in pharma background, how can we identify such things like wavelengths and all? How can we design such biosensors?
see, for that we actually did, and we got some papers on that. We first collected the data simulated in the lab, which took around 55 hours. Then we use those parameters to train and see whether it is giving for the graphene, for the MOS, which layer we are using, whether we are going with the gold or copper, whatever. So using that, initially we get it with some nanometers thickness and we got the results. Later we used a deep learning approach not to predict the highest FOM or which model, but to give it a range. Like I don't have to change the variations in both the wavelength and the thickness. I can just look at a particular range, a small range where I can get the highest FOM. So we use that work to reduce the search space.
Okay. Sir, means you have collaborated with any other department like physics or chemistry or some
Physics,
multi
I have
departments?
I have my scholar which is
Hmm.
working under the supervision of a professor from physics and under me from CSA. So this was the work which he was doing on FBG sensors.
Okay, okay.
Yeah.
Means we need to have idea on the materials also, like copper. You're saying
Correct, correct,
certain
correct. You need to have
materials.
idea because the person has to be a physicist.
Yes,
And
sir.
if a person is physicist, trust me, he will have very less knowledge about machine learning and deep learning. So we had one student who is a MTech from physics from IIT Delhi. Not exactly, but industrial engineering something was there, and he's working in ID domain. He has both the knowledge, so it became easy for us to define the problem statement.
Okay, sir. Thank you very much, sir.
Okay, ma'am. Okay. Thank you so much, everyone. Thank you so much.
Thank you, sir. We are grateful to you that you took time to talk to us.
Thank you so much.
And
I'll wind up.
do we have another query?
Shall we wind up?
Yes, sir. I was thinking that some participant was trying to speak and they were trying to say something. So I was not sure. Okay, okay. So if there are no more queries and all, we would like to conclude this session. And thank you, Dr. Bhadwaj, so much for talking to us. And it was a very informative session. So
Sure.
I would request you to accept this virtual memento on our part and as an acknowledgement for all your efforts. Thank you.
Thank you so much.
Thank you, Rishabh Singh.
Thank you, Rishabh sir.
Thank you,
Thank you.
Rishabh sir, uh, for all the a very dynamic and future-oriented talk on AI in healthcare. Thank you, sir.
Thank
Thank
you,
you.
sir. Thank you.
Thank you so much. Thank you so much. So we will be winding up for today. We'll be stop. The sessions will stop for today, and we will join everyone tomorrow again. So I would like to inform that the participants will have to fill all the feedback forms that have been shared in the chat box, and the feedback form will not be shared in the group. So all the participants who have been here should fill the feedback form from the chat box, and then only your attendance will be recorded for today.
Sorry.
So please fill the every feedback form that has been shared, that will be shared with you every day. So your attendance will be recorded on the basis of that. And on the end of the 6 days FDP, we will share an assessment link, and you will be required to complete that assessment and gain 60% marks in the assessment. So I hope you all had a knowledge filled and good learning day. So we look forward to you joining for more sessions tomorrow onwards again. Thank you. Have a great afternoon, everyone.